Hello everyone. Welcome to this tutorial on the embryological subdivisions of the human central nervous system. This tutorial pertains to our first core concept in the field of neuroscience, that the brain is the body's most complex organ. Although given the brevity and the relative simplicity of today's tutorial, as well as your progress in the course thus far, the complexity of the human brain won't exactly be front and center in our consideration. Really, what I'd like for you to be able to accomplish in this tutorial is to really refresh your appreciation and your understanding of the basic parts of the adult form of the human brain. And in so doing, what I want you to be able to do is to identify each of the major subdivisions of the adult nervous system. But now I want you to be able to relate them back to the embryological precursors and associated ventricular spaces. Well, after all, we're entering Unit 5 of medical neuroscience, and our goal is to understand the changing brain. And that story, of course, begins in embryological life with the first formation of the central nervous system that occurs really beginning in about the third week of the gestation of human life. And ultimately what we want to be able to do is understand, where did this marvelous form come from in the process of building the human organism? Well, I hope to simply review some of the parts that we see in the adult human brain with you in this session and relate them back to the terminology that we use to describe the developing brain in embryological life. So this will be largely review, and I hope that you'll appreciate this bit of a pivot as we turn from our studies of the adult form to a consideration of the mechanisms that are responsible for building the human brain, the body's most complex organ. So we will return to the lab and have a chance to once again review the anatomy of an actual human brain specimen. So I'll see you there. The lab lesson today is to recognize the embryological forms that are viewable in the adult brain. So let me reach into my bin and get a whole adult brain specimen. So when we look at the brain in its adult form, it's really amazing to think that this structure in front of me is derived from a simple tube. But as we just learned, at about the third week of gestation, the nervous system is in fact a simple tube. And that tube begins to form a very prominent bulge in the front part, and then two smaller bulges, one in the middle and one towards the back. And what we're looking at in front of us is mostly what has become of that forward bulge in that tube. And this is the cerebral hemispheres. This is the left hemisphere, this is the right. So that forward bulge divides into two parts. So most of what we see when we look at the adult brain is in fact this forward bulge. And we call this the prosencephalon, or forebrain for short. Pro meaning fore and encephalon meaning brain. So now we're looking at the posterior, the back side of the brain, and we can see a portion of the hindbrain, or the rhombencephalon. And the major structure that's visible here is called the cerebellum. The cerebellum is a large part of the brain which expands into two prominent hemispheres, a left hemisphere and a right hemisphere. And then if I just pick the brain up just a small amount, we can see the additional part of the hindbrain that's visible from the posterior surface, and that is the brain stem. But to really get a good look at the hindbrain, the rhombencephalon, we need to look at the ventral surface of the brain. So now we're looking at the underside, or the ventral surface of the brain. And we'll have another lab lesson on many of the features that we see. For today, I want to focus on the embryological divisions. So again, all of this cerebral cortex that we see in the hemispheres, this is part of the forebrain, or the prosencephalon. Now on the ventral side, we can see a small division of the prosencephalon that forms structures that we call in the adult form the thalamus and the hypothalamus. Together, this is called the diencephalon. And we can see that just behind this optic nerve and its crossing, there is a surface of the diencephalon here, that is the hypothalamus. And I believe we can see these two small bulges. These are called the mammillary bodies. And that is one surface landmark that tells us that we're looking at the floor of the diencephalon, the mammillary bodies being part of the hypothalamus. Well, just behind the mammillary bodies, we see the midbrain. And from this view, what we can see are two broad columns. These are called the cerebral peduncles. Peduncle is a word that means stalk, and these are two broad stalks that seem to support the cerebral hemispheres. That's the midbrain. Now, just below the cerebral peduncles is this broad surface here. This is called the pons. The pons is that part of the hindbrain that is intimately connected to the cerebellum. In fact, I think you can appreciate how the pons seems to stream out laterally right into the cerebellum on either side. So that's the pons. Together with the cerebellum, this is called the metencephalon, which is a subdivision of the hindbrain. And then finally, just below the pons is a complex part of the brainstem called the medulla oblongata, or just the medulla for short. And the medulla is that part of the brainstem that connects to the spinal cord. Now, from a different specimen, I'd like to show you what the human spinal cord looks like. And it would attach to this cut surface of the medulla. Now, attached to the bottom of the brainstem, of course, is the spinal cord. And I have several segments of spinal cord to show you. This is the upper third of the spinal cord that attaches to the bottom of the brain at the brainstem. But this has been removed, so I'll go ahead and set this brain down, and we'll turn our attention to the spinal cord. This is the upper third, which includes the cervical enlargement. And then this is the middle third of the spinal cord. That is the part that's in the region of the thorax. We call that the thoracic spinal cord. And then down below is the lumbosacral enlargement. The cervical enlargement is concerned mainly with our arms and our hands, and the lumbar enlargement is concerned mainly with our legs and our feet. And just below the lumbar enlargement is the most inferior segment of the spinal cord, and it's called the cauda equina. And as we get a close look at the cauda equina, you can see why it has its name. Cauda equina means horse's tail. And what we see is the terminus of the spinal cord with a set of spinal nerves that extend for several vertebral segments out from the vertebral column to serve the lower part of the body. Welcome to this tutorial on early brain development. This topic today will relate to several of our core concepts in the field of neuroscience. In this session, we're going to talk about how neurons communicate, but in a different context than what we did in Neural 2. So yes, indeed, neurons communicate using both electrical and chemical signals. But today, we're going to focus on the earliest events that led to the development of the nervous system. And we will explore in some detail a means of chemical communication that doesn't involve synapses, but it's cell-to-cell communication via chemicals nevertheless. And it is a mode of signaling that results in the acquisition of cellular identity, cellular fate, and the regional specification of structure in the developing brain. I think you'll find that a fascinating example of how specific molecules can be used to build a brain. Well, we will also be talking about the expression of specific genes in the developing brain. So, in a sense, we can apply actually core concept number 3, that genetically determined circuits are the foundation of the nervous system. We will not be talking about the formation of circuits in this tutorial. We'll save that for a later one. But we will definitely be exploring examples where the specific expression of genes is essential for building the foundations of the central nervous system. And then lastly, I think the core concept that I'd like to allude to, just briefly as we go through this tutorial, is that life experiences can change the nervous system. And the kinds of life experiences that I have in mind here are not the sensory motor experiences that we've been talking about in the last couple of units of this course, but rather the environment that the developing brain might find itself in, depending upon factors such as diet, the presence of environmental toxins. We can think of these as being very early life experiences that can shape the formation of the brain, unfortunately, often for the worse. Well, okay, with that as a bit of context, let's look at our learning objectives for this tutorial. First, I want you to be able to characterize the events that occur during some really important stages of early embryonic development called gastrulation and neurulation. I want you to be able to state the significance of neural induction. This is a really important process that I'd like for you to be able to understand and be able to discuss with a friend or a family member as a result of this tutorial. And neural induction is one of the key mechanisms that leads to the initial development of the central nervous system. Thirdly, I'd like to be able to have you discuss the factors that guide the migration of neuroblasts, which are the primordial cells that will give rise to the neurons and glia of the brain, and they have to migrate to get to their final destinations. So I want you to be able to have some capacity to discuss how that happens, how they migrate through the developing nervous system. I want you to be able to characterize the cellular mechanisms that influence the differentiation of neurons and glia. Obviously, they are distinct types of cells in the nervous system that serve distinct functions. And at some point in development, cells need to make a decision about what kind of cell they will become, a neuron or a glia. And we're beginning to gain some insight as to how that happens. And then lastly, in this tutorial, I want you to be able to describe the role of programmed cell death. We call that apoptosis in the development of the central nervous system. Obviously, we have an aggressive and ambitious agenda for this tutorial, so let's go ahead and get started. And I'd like to set the stage by talking in very broad terms about what I consider to be the 3 major forces that shape the developing brain. And I might even argue that these forces are critical for shaping the evolution of the mammalian brain, but that's a different topic for a different day. In the context of development, I want you to consider how these forces interact, and we will return to this theme as we progress on through our unit on the changing brain. So let me introduce these ideas and give you a sense of what I have in mind here. So when we think about the factors that shape the developing brain, I think a lot of us might come quite quickly to the notion of, well, okay, there's nature and there's nurture. But what do we really mean by these terms? Well, by nature, what I mean are lineage-derived signals that reflect the expression of specific genes. So these are genetic instructions, or what I like to call genetic specification. Okay, so nature reflects the genome that a cell has inherited from its parent cell, and there are specific patterns of gene expression that are responsible for much of the early formation of the nervous system. We'll spend a lot of time in this tutorial emphasizing this particular force in brain development. Well, as the developing nervous system acquires the functional capacity, then other kinds of developmental forces take shape. Well, we often think about nurture as something that might come a little bit later in brain development, as the developing nervous system acquires the capacity for sensory experience and motor behavior. Nurture can be thought of as environmental interactions, that is, experience-dependent modulation of brain activity. There's brain activity going on at later stages in development once synapses begin to form, and that activity is endogenous. That is, it isn't necessarily reflecting the interactions of sensory receptors with stimuli in the environment. But increasingly, as the brain does develop, and certainly in postnatal life, a topic we'll come to in a later tutorial, the experience of the infant becomes an important shaper of ongoing development of the brain. So we can think of nurture then as environmental interactions of the developing brain. And obviously, when we consider genetic specification and sensory motor experience, we recognize that these factors interact. Well, until fairly recently, I probably would have been ready to move on at that point. But thanks to some wonderful collaborators that I've worked with over the last few years who come from a very different discipline. My collaborators, my friends, they're actually theoretical physicists. And they've helped me understand that there's really a third major force in brain development that biologists ought to confront. And so I've been happy to be educated and to really embrace this idea and actually make it part of my research program. And that is to recognize that self-organization is a very important force that shapes the development and the maturation of circuitry in the central nervous system. So what do I mean by self-organization? Well, self-organization reflects the interactions of one cell to another, or many cells with respect to their neighborhood environments. And these interactions are mediated via activity-based mechanisms. So self-organization reflects the dynamical systems property of the developing brain. So as systems form synaptic connections and they acquire the capacity to generate electrical signals, and activity now becomes possible, self-organization begins to shape the distribution of synaptic connections and axonal arbors and the structure, what we call the functional architecture of neural networks. And I've been slow to come to appreciate this concept because until fairly recently it hasn't really been possible to apply quantitative tools to characterize the organization of circuitry in the brain. But now that we can do so, it's becoming really quite compelling, I think, that the structure and the form and the precision of synaptic connections that we see in neural networks is far greater than what can be accounted for from the genome, from genetic specification. And we've done some experiments, and others I think would concur that the degree of specification of circuitry in the brain couldn't possibly be instructed by sensory motor experience. So there must be an additional force, additional factor at play. And that additional factor is self-organization. And so self-organization interacts with sensory motor experience, it interacts with genetically encoded mechanisms, and together these three forces shape brain development. Now, as you might imagine, as these forces interact, there's opportunity, unfortunately, for things to go awry. Well, there are various ways in which these basic mechanisms of development can go awry in brain development. There can be mutations of genes that affect how these genetic instructions are read out, how the genes are transcribed, and how the messenger RNA molecules are translated into proteins, and then how those proteins are actually processed and inserted wherever their functional role happens to require their presence. There are also interactions among cells that are mediated via these activity-based mechanisms that are responsible for self-organizing networks in the brain. And while self-organization seems to be remarkably robust against perturbations, one can imagine that the self-organization of the brain might be forced into some different kind of state or some different kind of dynamical system, depending upon the interactions perhaps with molecules present in the environment that might affect how genetic instructions are played out. And then lastly, one can imagine that sensory motor experience could be important in later stages of brain development, and indeed perhaps across the lifespan, as the experiences of the body begin to feed back upon the structure and function of the human brain. And I think what we've learned from a variety of sources is that sensory motor experience can cut both ways. It can be beneficial, it can promote the maturation of the nervous system, or it can be detrimental. It can reinforce functional impairment. Well, these are topics that I want to progressively unfold, but I do want to get them out there in front of you as we begin this consideration of the development of the nervous system and how the nervous system changes across the lifespan. All right. So what I'd like to do now is talk about the earliest stages of the formation of the nervous system, which will largely reflect the expression of genetic specification in the developing brain. Well, the story of the developing nervous system really begins with this process that we call gastrulation. So gastrulation refers to an invagination of the developing embryo at a stage that we call the blastula. And this process of invagination in the blastula produces three principal germ layers. And those germ layers are the ectoderm, the most outer layer, the mesoderm or the mesoderm, which is the middle layer, and then the endoderm, which is the inner of these three layers. This is a fundamental event in the developing embryo, and it sets the stage for the formation of the nervous system. Now, one very important structure that is formed in this stage of development is something called the notochord. Now, the notochord is a long rod-like structure that is derived from mesoderm. And here, if we make a cross-section through this developing embryo, we see the notochord here in this central location cut in cross-section. So what's special about the notochord? Well, I'll say more about that as we progress and talk about inductive signaling. But the notochord is responsible for sending out chemical signals that interact with this ectoderm that overlies this rod-like structure that runs along the length of the embryo. And these chemical signals induce a change in the fate of the ectoderm that overlies this notochord. That region of this ectoderm differentiates into a columnar epithelium that we call the neural plate. And this neural plate, or sometimes we call it neuroectoderm, will begin the process of forming the entire central nervous system. So the notochord is responsible for inducing the differentiation of the neural plate. I should also mention that the notochord helps to establish our primary axes of orientation in the developing embryo. The notochord forms in a more dorsal position in the developing embryo. Now we'll call it a gastrula. And so the presence of the notochord helps us define what is dorsal versus what is ventral. The length of the notochord establishes the anterior to posterior axis of the developing embryo, and the fact that it's a midline structure helps to define the axis from medial out to lateral, obviously in both directions. So the presence of the notochord helps to define the bilateral axis of symmetry in the developing nervous system. Well, following gastrulation and the development of the notochord and the onset of inductive signaling that forms the neural plate, the next major phase of embryogenesis is called neurulation. Neurulation is defined by the initial formation of the nervous system. And what happens in neurulation is that the neural plate begins to rise up in its lateral margins and begins to close off in a process that will form a tube out of that neural plate. So this begins with the formation of a groove that runs the anterior to posterior length of the embryo, just above the notochord. And as the walls of this neural tube begin to grow out, they begin to come together along the dorsal midline. And we can see that process transpiring over weeks 3 into week 4 in human embryogenesis. Here we proceed a little bit further, and we can see that now the neural tube has actually closed off underneath this overlying ectoderm. And there is a progressive closure of this neural tube that begins near the center of this developing neural tube and then extends out into the anterior and posterior directions. So this process continues, and now we're well into the 4th week of embryonic life, and we have the anterior end and the posterior end of this developing neural tube progressively closing. The anterior end of this developing neural tube is going to form the brain, and the rest of its length, all the way down to the posterior tip, is going to form the spinal cord. Now, I'd like to return to this concept of inductive signaling and say a little bit more about it, because it's at this stage in embryogenesis neurulation that the inductive signals derived from the notochord are so important for defining many of the important regions of that developing neural tube that will set the stage for further differentiation into the brain, into the spinal cord. So if we back up just a little bit in neurulation, so over here to the left we have the folds of the neural plate beginning to come together to form the neural tube, and the notochord is giving rise to these chemical signals that are inducing these morphogenic events in this overlying neural plate. But in addition to simply inducing the folding of that neural tube, well, I shouldn't say simply, it's really an amazing feat in development that such a thing would happen. But in addition to inducing the formation of this tube, the notochord is also giving rise to signals that are specifying some particular regions. And one particular region is that portion of the neural plate that directly overlies the notochord. This becomes a region that we call the floor plate. And this becomes quite important because it in turn gives rise to inductive signals that will influence the surrounding portion of this neural tube. Now there's another special region that responds to inductive signals, and that is found on the dorsal side of this developing neural tube. We call that the roof plate. And the roof plate will give rise to signals that will influence the dorsal aspect of this epithelium that is forming the walls of the neural tube. And one additional region I want to highlight for you is that in the ridge of this folding neural tube that begins to come together, there's a special population of cells here that we call the neural crest. And the neural crest will actually pinch off from the margins of this neural plate as the neural tube is forming. And the neural crest ends up sitting just along the dorsal and lateral margins of the neural tube once that tube has formed. Now the neural crest is a source of many different types of cells that are derived from it. Cells begin to differentiate and they begin to migrate away from this region of the neural crest. And as they migrate away, they are subject to chemical signals from the nearby structures in the mesenchyme and in the mesodermal derivatives, such as the somites that we see here sitting just lateral to the neural tube. So as these cells migrate away from that dorsal lateral neural crest, they are exposed to all kinds of signals that generate a rich variety of progeny of these neural crests. And these progeny include both neurons as well as non-neural structures. So here's just a brief example of some of the derivatives of this neural crest. So the neural crest progenitor cell can be influenced by various factors to form our sensory neurons that we find in structures like the dorsal root ganglia. Other signals can induce the formation of the peripheral cells of the visceral motor system. And so that would include our ganglionic neurons that are innervating our visceral tissues. So essentially, the entire peripheral nervous system, including the enteric nervous system, is derived from neural crest. And as I mentioned, also non-neural structures are derived from neural crests, such as the chromaffin cells and the melanocytes, and others that are not depicted here. Now, how is all this accomplished? How is this possible? Well, it is possible because of inductive signaling. If I can just back up to this slide again and just highlight the means by which these signaling interactions occur. So I mentioned the notochord is a very important source of inductive signaling. The floor plate and the roof plate become important sources of inductive signaling, as do the somites and these mesenchyme cells through which the neural crest derivatives must migrate. So, just to be clear, I've highlighted for you in the handout what I mean by inductive signaling, so let me just draw your attention to that. So inductive signaling is the ability of a cell or a tissue to influence the fate of nearby cells during development by the synthesis and secretion of chemical signals. Now, these chemical signals are either steroid hormones or peptide hormones. So they have an impact on the transcription of genes in the cells that they interact with. So the precise timing of the expression of these inductive signals becomes crucial for the proper formation of the developing brain. So what we will see is that in order to form the nervous system correctly, these inductive signals need to be turned on and turned off in a very precise pattern in both space within the embryo, but also in terms of developmental time. And all of this is regulated in a complex way that we're just now beginning to understand. And again, it's subject to perturbation that can produce various forms of defects in the developing brain, as inductive signals might be either blocked or perhaps overexpressed. So we'll see just a few examples of that as we go along. But now what I'd like to do is to turn our attention to the large-scale events that account for the structure of the adult brain as now you've come to understand it as we've worked through the early units of this course. So, how is it that we can go from a simple tube to this marvelous structure that you've now come to appreciate as the adult form of the human brain? It's hard to look at this, is it not, and imagine that at some early stage in development, this brain was a simple tube, but indeed that's the case. So what I'd like for you to be able to do is to reflect upon what you know about the anatomy of the adult form of the human brain and relate it back to the more simpler forms that are produced in embryonic life. Okay, well, to get there, let's just look at the development of the brain and the spinal cord in the very broadest terms, as if we were looking at this from a fairly macroscopic perspective. Well, what I'd like to show you are a series of illustrations that show the various transformations of regions of that neural tube that give rise to specific parts of that adult human brain that you now know something about. Well, over to the right, we have an illustration in schematic form of our neural tube, and so it's a fairly simple representation of that tube, but we're really only showing the anterior end of that tube. To the left, we have a picture of the developing embryo. So here is the embryo at a fairly early stage in development, probably about 4 weeks or so, 5 weeks. And what we find here is that the developing embryo has a shape that looks something like a cane, as our textbook describes. I hope you're not offended by that. This is vertebrate life, after all, and a representation of human life. But what we find is a shape that reflects some morphogenic events that begin to take place in the anterior end of this developing neural tube. What I mean by morphogenic events is the differential proliferation of cells that cause this simple neural tube to begin to bulge in certain places, to bend in others, or to constrict. And what we find is the formation of essentially three principal enlargements in the anterior end of this neural tube. The most anterior of these enlargements we call the prosencephalon. So the prosencephalon can be found here at the anterior end of the neural tube, and if we see this cane-like shape, it's at the end of the handle of the cane, so right in this region. And this prosencephalon is what will go on to form the cerebrum, the hemispheres of the brain. So it's the front brain, the first brain, that's why we call it the prosencephalon. So just behind the prosencephalon, we have the middle of these three swellings, and we call that the mesencephalon, which means middle brain. So this middle brain will go on to form what we call the midbrain. And then lastly, the third of these swellings, just caudal or towards the tail, behind the mesencephalon, is called the rhombencephalon. And the rhombencephalon is what we call the hindbrain. So the rhombencephalon would be this relatively extended straight part at the top of the cane-like shape of this developing embryo. And then caudal or posterior to the rhombencephalon, we have the spinal cord, which will take shape. So these three principal divisions go on in development, and they begin to further subdivide, specifically the prosencephalon and the rhombencephalon. So the prosencephalon begins to divide into two principal divisions, a diencephalon and a telencephalon. The telencephalon will begin to form these large vesicles that will continue to expand and dominate the developing nervous system. So these telencephalic vesicles are what will give rise to the cerebral hemispheres. The diencephalon, meanwhile, undergoes its own interesting changes. The diencephalon will begin to form these outpockets that we call the optic vesicles. And the optic vesicle is what will go out to form the neural elements of the eye, specifically the retina. And of course, as I mentioned when we study the visual system, the retina can therefore be seen as really a part of the brain. And the connection between the retina and the rest of the diencephalon really reflects its embryological history. This is why the optic nerve is myelinated by oligodendrocytes, the glial cell that makes myelin in the central nervous system. And this is why, in one perspective, we might argue that the optic nerve really isn't a nerve at all. It's an extension of the brain. Okay, well, let's move on and consider briefly what happens to the mesencephalon. So the mesencephalon is that middle brain. This is the division of the developing neural tube that seems to not change so much relative to the others. It doesn't necessarily increase in size proportionately, nor does it differentiate or subdivide any further in any significant way. So just caudal to that mesencephalon then is the hindbrain, the rhombencephalon. And the rhombencephalon actually does begin to show some interesting differentiations. There begins to be a bend at the top of that cane-like handle, and that we call the pontine flexure. And this flexure begins to differentiate two principal components of the rhombencephalon. In the more rostral direction, we have a metencephalon, and then more caudally, just posterior to this flexure, is the myelencephalon. So the metencephalon is what will go on to form the pons and the entire cerebellum. The myelencephalon will form the medulla oblongata. So let's advance one more stage in embryogenesis. So now we're well into the second trimester of gestation, and we begin to see a nervous system that resembles what you've come to appreciate in the adult form. So the view of the developing brain at this point is obviously dominated by the cerebral hemispheres. So this is, of course, the telencephalon. And if we were to look at this forebrain in cross-section, what we would see is not only an outer cortex developing in this telencephalic vesicle, but also we would see deep gray matter. And that deep gray matter, of course, is the basal ganglia. Well, not seen from the outside, but present and visible in cross-section, is that derivative of the prosencephalon that is immediately caudal to the telencephalon. And what I'm referring to, of course, is the diencephalon. And that diencephalon is beginning to differentiate into a thalamus, which is found more dorsal, and then not seen at this level, more ventral and a little bit anterior would be the hypothalamus. So from the diencephalon, we get the thalamus and the hypothalamus. And then if we looked a little bit deeper, also not shown in this particular cross-section, we would see the mesencephalon, which is now completely dwarfed by the expansion of the prosencephalon into the telencephalon and the diencephalon. And just caudal to that mesencephalon, we would see next the derivatives of the rhombencephalon, or the hindbrain. And those derivatives would be most obviously the cerebellum, and that portion of the brainstem to which it's most intimately connected, and that would be the pons. Together, the pons and the cerebellum then are the metencephalon. And then we would begin to see a nicely shaped myelencephalon, which is a transitional region between divisions of the brainstem and the upper segments of the spinal cord, which is, of course, what then extends down the vertebral column of the developing fetus. Well, what I'd like for you to be able to do is to tell this story of the major events that shape the formation of the brain, and to be able to relate the adult brain structures that you've now come to recognize and know something about their structure and their physiology. I want you to be able to relate those derivatives back to the embryonic brain. And I want you to include in that discussion the associated ventricular spaces. So hopefully, as we begin to put together your understanding of the anatomy of the human nervous system, this embryological framework will actually come to help you. So for example, if you are looking at a cross-section through the brain and you're not quite sure what you're looking at, perhaps you will remember that what you're seeing near the dorsal aspect of the cross-section is actually a very narrow channel, allowing for the flow of cerebral spinal fluid from the third to the fourth ventricle. You may recall that that's the cerebral aqueduct, and that would be a hallmark of the midbrain. So seeing the cerebral aqueduct is one way to identify what kind of section through the brainstem you might be inspecting. Well, having an understanding of embryogenesis helps you appreciate why the lateral ventricles show up where they do. The lateral ventricles are present both along the dorsal aspect of the brain, but also down into the temporal lobe, and even extending back into the occipital lobe. And the development of the temporal lobe is an important aspect of human brain development. And I think what you have come to appreciate is that there are certain structures, in addition to the lateral ventricle, such as the caudate nucleus, a component of the basal ganglia, that are pulled down and anteriorly into that developing temporal lobe. So having this embryological framework, knowing where things came from, can be a great aid to you in understanding your appreciation, your perspective of human brain anatomy. Well, if you've not done so yet, I would encourage you to view my brief tutorial on the embryological divisions of the human brain, and to use this chart as a way to reflect upon what you're seeing as That video tutorial brings you back to the lab and shows you the human brain and allows you to recognize the various subdivisions that I've been talking about as they actually present in an adult brain specimen. So if you've not done so, go ahead and do that at this point, and we'll resume our discussion in this tutorial after you've had a bit of reinforcement of your ability to recognize human brain anatomy in the adult form and be able to relate that back to the embryological origins. Well, now that you've been able to refresh your understanding of the anatomy of the adult form of the human brain in relation to the changes that took place in the neural tube that gave rise to this fantastic form, what I would like to do is to begin to talk about some of the genetic and molecular mechanisms that are responsible for establishing regional identity along that developing neural tube that ultimately leads to the formation of the human brain. Well, this story really took off as biologists began to explore the genetic basis of development in invertebrate systems. And of course, one of our favorite invertebrate model systems in biology is the fruit fly Drosophila. And what was discovered is that there are a set of genes that help to define the segments of the developing Drosophila larva. So here is a representation of the Drosophila larva, and what we appreciate, of course, is that this animal has a segmented body plan. So what was discovered several decades ago is that the segmentation of this developing larva reflects the expression of particular genes that establish regional identity, such as this first thoracic segment of the larvae, which goes on to form what we call the prothorax in the Drosophila. And it's from that prothorax that particular body parts emerge, such as this first limb that emerges from the transformed larva into the adult form. Well, much molecular and genetic biology has gone on over the decades, and we've come to recognize that this segmentation of the developing larva into an adult form reflects the expression of what we call in Drosophila Hox genes, and their homologs in mammalians, including humans, are called homeotic genes. So in the human genome, we now know that there are 4 clusters of these Hox genes, or these homeotic genes. Sometimes we call them homeobox genes. And these clusters are responsible, we think, for beginning to establish some regional identity in the developing nervous system. There is an anterior to posterior pattern of expression of these clusters of homeotic genes that help to establish, for example, the various segments of the human spinal cord and the major divisions of the brainstem. Well, why should this be important? Why should you be motivated to learn something about these homeotic genes? Well, I've already challenged you to consider in some detail the anatomy of the differential regions of the human brainstem, for one very good reason, to understand the location of the cranial nerves in relation to divisions of the brain. So hopefully you recognize the relation of the trigeminal nerve to the pons, and at the junction of the pons and the medulla we have an abducens nerve, which is not present in this particular specimen, but we see very nicely here are cranial nerve 7, the facial nerve, and certainly a very beautiful vestibular cochlear nerve present, cranial nerve 8, and even just, you can see, I think here, just a few nerve roots of cranial nerve 12 between the medullary pyramids and the olive, which is present out here laterally. Well, the relationship of these cranial nerves to particular regions of the developing brainstem seems to reflect the specific expression of homeotic genes that help to identify regional identity along the length of the developing neural tube. This is especially important for the specification of position within the developing brainstem. we see the expression of particular motor nuclei of the brainstem, for example, that seem to be derived from particular regions of the brainstem that express a set of these homeotic genes. So this is an example of genetic specification determining regional identity, and there are probably inductive signals at work here that establish this identity. And one of the many outcomes of this kind of inductive signaling is the specification of the identity of neurons that go on to form critical structures that are important for form and function in the human nervous system, including these cranial nerves. So if you've wondered why are the nerves found exactly where they are in the human brain, part of the answer is going to have to do with the differential expression of homeotic genes at particular points in development and at particular locations along the length of the developing neural tube, especially in that region that goes on to form the brainstem. Well, next I'd like to turn our attention to this matter of inductive signaling and give you a few examples, not to overwhelm you with molecular detail, but rather to give you a feel for how inductive signaling works and how these signals that are secreted out into the extracellular spaces in the developing embryo can have such a formative impact on the fate of cells that are in the process of developing within the walls of the neural tube. Well, as I mentioned, inductive signaling really begins in earnest in neurulation as the neural tube begins to form, and signals are expressed by this notochord that begin to influence the differentiation of these special regions in the neural tube that establish a dorsal ventral axis. There is a floor plate that begins to give rise to its own signals that will establish a ventral identity in this region of the developing neural tube. And I think you know enough now about the spinal cord to connect the dots here and realize that ventral identity is going to give rise to motor circuits and alpha motor neurons that will grow out and connect the central nervous system to effector systems, namely our striated muscles. Meanwhile, this roof plate will give rise to inductive signals that establish a dorsal identity in the developing walls of the neural tube. And dorsal, I think, as you reflect on your understanding of the spinal cord, implies the development of somatic sensory neurons that are receiving incoming information about the experiences of our peripheral tissues and integrating them within the central nervous system, giving rise to local circuit connections as well as long-distant pathways that convey somatic sensory signals to more rostral regions of the developing brain. So dorsal and ventral become really key regions within the developing neural tube that have an influence on the ultimate fate of the cells that are developing in those regions. So I'd like to give you some specific examples of some of these inductive signals so you have a bit of a feel for really what's going on here. So one of the best understood inductive signals is called retinoic acid. So retinoic acid is a small lipophilic molecule metabolized from vitamin A. It's a very important inductive signal, and it's one of the principal substances that explains the importance of vitamin A for early brain development. So retinoic acid is synthesized and released, and as a lipophilic molecule, it can readily translocate through cellular membranes, and it can bind to a receptor within the cell, and that receptor becomes a transcription factor that translocates to the nucleus, and it can interact with other binding proteins and turn on particular genes. And so that's what we see here. We see retinoic acid interacting with its receptors and other complexes that can modulate gene expression. Now, retinoic acid provides one of those examples where the life experience can have an impact on the developing brain insufficient vitamin A in the diet of the mother can have an impact on the capacity to produce this important inductive signal, and there can be deleterious consequences for the formation of the early nervous system. There is an additional problem that can be encountered with respect to retinoic acid signaling, and that is an excess of vitamin A. And there are a variety of dietary supplements and even topical agents that can be purchased for other reasons that include vitamin A or retinoic acid, and excess retinoic acid in the developing embryo likewise can be extremely harmful. In fact, retinoic acid can become what's called a teratogen, which is a exogenous substance that can induce malformations in the developing embryo. If you'd like to learn a little bit more about this particular topic, I would refer you to box 22C found in our textbook, which gives you a little bit more of the backstory regarding how retinoic acid signaling shapes the developing brain. Well, let's consider some other kinds of inductive signals. Most of our inductive signals are actually not steroid hormones, they're peptide hormones. So these are substances that are secreted and interact with surface-bound receptors. And one of our peptide hormones that I'd like to talk just a little bit about is called bone morphogenetic protein, or BMP for short. And the way BMP works is that it interacts with a surface receptor. Here we see BMP being secreted by one kind of cell, and it interacts with this receptor. And the receptor is what we call a serine kinase. So this means that it phosphorylates a serine residue in target proteins. And this serine kinase phosphorylates a transcriptional regulator called SMAD, or SMAD for short. This is an acronym. You don't need to worry about its name, but I do want you to understand something about how this system works. So the activated SMAD then associates with additional helper proteins, and this then translocates to the nucleus, where it acts as a transcription regulator. Now, BMP signaling is especially important in mesodermal tissue. As the name implies, bone morphogenetic protein, BMP signaling can induce the development of bone cells. Now, in ectoderm, BMP signaling can induce the formation of epidermis, or skin, unless BMP signal is controlled. And this is where these additional factors that are illustrated here come in. There are factors called noggin and chordin, they have somewhat fanciful names, that can antagonize the interaction of BMP with its receptor. When noggin and chordin antagonize the BMP signaling pathway, preventing BMP from interacting with its surface-bound receptor, that ectoderm will be diverted from an epidermal fate to a neural fate. With noggin and chordin, this tissue will differentiate into what we call the neuroectoderm, or the neural plate. Now, I think this is a really cool and fascinating thought, which I've highlighted for you in your tutorial notes, and that is that what these molecules, noggin and chordin, really do is they rescue this ectoderm from becoming skin. They induce the differentiation of this neural plate and subsequently the formation of the entire central nervous system. So from that point of view, I think we can all thank our noggin and chordin for giving us a brain and a spinal cord. One last inductive signal I'd like to just talk through is one that has perhaps the most memorable name of all, especially those of you that have some history in video gaming. This inductive signal is called SHH, or Sonic Hedgehog. And Sonic Hedgehog is a protein hormone that interacts with receptors that are bound to the surface, and its receptor is a protein called patched that interacts with a protein called smoothened. And these interactions will then go on to activate a series of transcription factors that were originally identified in brain tumors or gliomas. And when Sonic Hedgehog interacts with Patched in the presence of Smoothed, what we find is a switch in the transcription factors that are regulated within the cell. And specifically, this GLI1 becomes induced, and it binds to DNA and modulates gene expression. This Sonic Hedgehog mediated signaling pathway is critical for the proper closure of the neural tube. Well, just to look at the big picture now of inductive signaling, and this is a slide from the book that gives you way more detail than I care for you to know in this course, but it does represent a paradigm that I want you to appreciate. And that paradigm is what we might call a transcriptional code, which reflects the coordinated expression of inductive signals that are coordinated both in location but also in developmental time. And this interaction of inductive signaling is what gives rise to regional identity within the developing nervous system. It helps to differentiate one location in the brainstem or spinal cord from another. It also establishes the dorsal ventral axis of the developing neural tube, which is critical for the development of sensory neurons, as well as columns of cells that give rise to our motor output from the brainstem and the spinal cord. So these inductive signals really provide for the establishment of the identity of distinct populations of neurons that are necessary for the expression of mature function in the human brain and spinal cord. Now, a similar transcriptional code is responsible for the formation of regions that go on to form our forebrain, our telencephalon and our diencephalon. We know quite a bit less about how this works in the forebrain than in the hindbrain and the spinal cord, but we're beginning to understand some of the inductive signals that are responsible for forming the cerebral cortex, the basal ganglia, other deep forebrain structures, as well as the derivatives of the diencephalon, including the optic cup and the otic placode, which goes on to form the structures that are necessary for vestibular function and auditory function. Well, once regional identity has been established along the length of the developing neural tube, the next major challenge is proliferation, making the cells that are necessary to properly populate the gray matter structures of the developing brain and spinal cord. And proliferation is just mind-boggling in the developing nervous system. We know that in the adult brain, there are about 100 billion neurons. And if that number doesn't stagger the mind, consider that there are at least that many, and probably several fold greater than that number, glial cells in the human central nervous system. So just an incredible number of cells are present in the brain and the spinal cord. And in order to produce so many cells, the rate of proliferation is just staggering. During the peak of the production of cells in the developing nervous system, scientists have estimated that perhaps as many as 250,000 cells are produced every minute in the developing embryo. Another amazing aspect of the proliferation of brain cells in early development is that the production of neurons is restricted in time to a very narrow period. Nearly all of the neurons that we have in our central nervous system right now were born in the second trimester of pregnancy. Now, there are some brain structures, like the cerebellum, that has just an enormous number of cells, and I'm referring to the granule cell population of the cerebellum, and it takes a considerable longer period of time to populate this cerebellum, so at least a year, perhaps as long as 2 years. But with the exception of the cerebellum, virtually all of the cells that we have in our brain and spinal cord were born in that second trimester of pregnancy. Now, some of you may know that there is adult neurogenesis. That is, even right now, there are new neurons being born. But, as we'll come to in a few tutorials, the genesis of those new neurons is restricted to the hippocampus, at least in our brain. And it is perhaps very important that this phenomenon occurs. It may contribute to learning and memory and other aspects of maintaining our mental and physical health. But it is a restricted population of cells that grow into that one region of the brain. And as far as we know now, the impact of the stem cells that may reside in the human brain seems to be limited, both in terms of normal physiological function and the response of the brain to injury. So I think there are fairly profound implications for this restriction of neurogenesis in the formation of the nervous system. The neurons that we have right now are the ones that were born in that second trimester of pregnancy. So I would encourage you to think about that and wonder, well, what are the implications for learning and memory? What are the implications for rehabilitation following injury, if that's the case? Maybe that's something that we can kick over to the discussion board and see if that might inspire some interesting dialogue. Well, let's talk a little bit about how this proliferation of brain cells occurs, and then we can go on to talk about how brain cells are differentiated into neuronal or glial fates. Brain cells, like most other cells in the body, go through the usual stages of growth and the synthesis of new DNA, setting the stage for the division of the cell into daughter cells. And that division occurs via a process that we call mitosis. I certainly am assuming that you are broadly familiar with cell division from your prior studies of cell and molecular biology. So I won't go into the details of how mitosis occurs. Rather, I'd like to talk about how mitosis happens in the walls of the neural tube that allows for the production of the cells that come to populate specific gray matter structures that we recognize in the human brain, such as the cerebral cortex or the basal ganglia. Well, proliferation occurs among a population of precursor cells in the walls of the neural tube. These precursor cells have a recognizable cell body containing the nucleus and all the other organelles that are common to the cells of our body. And that cell body then grows a process that makes contact with the inner and the outer surface of this wall of the developing neural tube. The outer surface will become the peel surface of the brain, and the inner surface is, of course, the inner surface of the neural tube that goes on to differentiate into the ependymal lining of the ventricles in the human brain. Now, along this path between the peel surface and the ventricle surface, this cell body becomes motile. That is, it migrates in the up and down directions towards the pia and then back towards the lumen of the ventricle. So as this cell body begins to translocate up towards the peel surface, the cell now enters the synthesis stage, where DNA is being replicated. And then as DNA is replicated, the cell body then migrates back down towards the luminal surface of this developing neural tube as the cell gets ready for division. And it's near the ventricular surface then of this neural tube that mitosis occurs. And mitosis can happen in a symmetrical way. It can produce two precursor cells that can go through this dance of migrating to the peel surface and back to the luminal surface again, and undergo subsequent cycles of mitosis, producing more daughter cells. Or there can be what's called asymmetrical division. An asymmetrical division produces two different kinds of cells. One cell remains a progenitor cell, which can go on to subsequent stages of mitosis, although its mitotic potential now is somewhat limited. The other daughter cell becomes what we call a neuroblast. And this is now a postmitotic cell that can go on to differentiate into neurons or glial cells. Now, once this neuroblast has been produced through this process of asymmetrical mitosis, for many of these cells, they migrate away from the ventricular region of the wall of the neural tube to a more peripheral destination. And that migration is important for forming a structure like the cerebral cortex. So now we're looking a little bit further along in development, and now the wall of the neural tube is beginning to become a bit more complex. And if we look at this wall of the neural tube in some detail, we can recognize the ventricular zone where mitosis is happening. But increasingly, we begin to develop a separation between the peel surface and the luminal surface in the wall of the neural tube. And we begin to populate a structure that now acquires the properties of gray matter. And this structure we call the cortical plate. The cortical plate is the first formation of the cerebral cortex. So the challenge here is to get these neuroblasts that are formed here in the ventricular surface up to the cortical plate. And this requires a process of migration. So the cells must become motile, and they must find their way to this outward cortical plate. And the way this happens is these neuroblasts will migrate along a radial fiber that is formed by a presumptive glial cell. We call this the radial glial cell. So these are essentially precursor cells now that continue to maintain their contact between the peel surface and the luminal surface of the developing neural tube. And this long extended process becomes a pathway for the migration of the neuroblast. So over here to the right, we have a depiction of one of these neuroblasts migrating along this radial glial fiber. And these cells will translocate. They will shimmy along this fiber on their way towards the cortical plate. So there are a variety of factors that guide this migration, and along the way, these neuroblasts begin to further differentiate and acquire a specific identity that will allow them to be differentiated into a neuron or a glia or a particular subtype of one of those broad categories of brain cells. So as these neuroblasts migrate from the ventricular zone up to the cortical plate, they do so in a particular pattern. The cortex is essentially built in an inside-out fashion. And here are some data from what's called birth dating studies that have been done to track the final mitosis of a precursor cell and its ultimate position within the developing cortical plate. Well, there's a technical means by which this can be done. It's described in a box in Chapter 22 if you're interested in how this happens. But essentially, what we can do is we can label precursor cells at a particular point in development and simply ask what has become of the postmitotic neuroblasts that were derived from that population of precursor cells that were marked at a particular time in development. And what this approach has taught us is that the cortex does indeed develop in an inside-out fashion. The cells that are born in, let's say, the late first trimester of pregnancy come to populate the inner and the outer margins of the cortical plate. And thereafter, as we progress through the second trimester of pregnancy, the cells that are born early in that second trimester come to reside in the lower portion of the cortical plate, in a region that ultimately differentiates into cortical layer 6, the inner layer of the cortex that sits just on top of the white matter. And as neurogenesis progresses through that second trimester of pregnancy, extending at least in some cortical regions into the third trimester, we see this progressive population of the cortical plate from the inner layers to the outer layers, such that the last cells to take up their residence in the developing cortical plate are those that reside in what will become cortical layers 2 and 3. So these neurons that come to reside in the outer part of the cortex have to migrate through these cells that have already established residence in the deeper layers of the cortex. And as these cells migrate through these deeper layers of the cortex, there is yet additional opportunity for inductive signaling to influence the fate of these later born and more superficial residing cells in the developing cortex. So this is the pattern by which a cortex is constructed, an inside-out pattern involving the precise regulation of neurogenesis and the migration of neuroblasts. So obviously there's a lot of specification that's going on here. Cells are born at a particular time. They are provided the signals that are essential for migration and for getting off that radioglial fiber at the appropriate location, such that the cortex is progressively built out in this inside-out pattern. Now, unfortunately, we know that some of the signals that are responsible are subject to genetic mutation. Perhaps they can be influenced by environmental toxins or dietary factors. And there are some known phenotypes where this inside-out pattern is disrupted. In fact, the process of neuronal migration and the proper establishment of this inside-out pattern seems to be essential for establishing the identity of the cells that take up residence in these various layers of the cortex. And their location, as you may recall, has much to do with determining the connections of the neurons that are ending up in one cortical layer or another. So if this process of migration and patterning of the cortex is disrupted, it's very likely that also the connections of those cells will likewise be disrupted. And we now know that there are a number of congenital malformations of the cerebral cortex that reflect errors in the migration of these neuroblasts. We think that perhaps even subtle errors in the migration of these neurons might be responsible for some forms of mental retardation and learning disability. Although there's much more work that needs to be done to truly understand what's going on at the level of cortical development that can be related to the broad spectrum of learning disabilities that we confront in society. We'll say more about that topic as the story of cortical maturation continues in subsequent tutorials. Well, this inside-out patterning of neurons that come to populate the cortical plate is largely a story of the genesis of our pyramidal cells, that is, the principal projection neuron of the cerebral cortex that produces glutamate and uses that as an excitatory neurotransmitter in the brain. Well, we know that the cortex also has inhibitory interneurons. And for many years, we just assumed that the inhibitory neurons of the cerebral cortex follow the same kind of inside-out patterning being derived from neuroblasts that migrate from the ventricular zone up to the developing cortical plate. Well, I think it was a big surprise to many of us when it was discovered that that's actually not the case, that most of the inhibitory interneurons that come to reside in the cerebral cortex are actually derived from a developmental region that's deep in the base of the forebrain. We call this region the ganglionic eminence, and it differentiates into a lateral and a medial division. And it's from these ganglionic eminences that inhibitory neurons are born and differentiate. And as they differentiate, they migrate, not just in the radial dimension, but also in the tangential dimension. So it's from the ganglionic eminences that our inhibitory interneurons are derived and come to populate the cerebral cortex through this process of tangential migration. There are other examples of tangential migration in the nervous system that affect other kinds of cells. You can read more about that in the textbook if you'd like. But for this particular point, I would just want you to appreciate the fact that our pyramidal cells are derived from the ventricular zone and migrate in a radial dimension to reach the cortical plate, while our inhibitory neurons are derived from these ganglionic eminences deep in the forebrain, and they migrate, in some cases, quite a long distance in the tangential direction to reach their ultimate destination in the cerebral cortex. And the implication would be that there must be signals that guide their migration that inform those neuroblasts about what their ultimate destination must be. Well, the ganglionic eminences also give rise to the basal forebrain structures, including the basal ganglia and some cells that are part of the amygdala formation. So in addition to these inhibitory neurons, this part of the developing prosencephalon gives rise to those telencephalic structures that we consider the deep gray matter of the forebrain. So we've talked now about the proliferation of cells that give rise to the neuroblast and how they migrate and come to populate specific gray matter structures like the cortical plate and the basal ganglia. So one important question that I hope you've thought about at this point in the tutorial is, how is it that these precursor cells that are postmitotic and migrating to a particular location come to differentiate into the diverse forms that we recognize among our neurons and our glial cells that we discover in the adult brain? Well, there are basically two broad kinds of mechanisms that explain the differentiation of these precursor cells into their final fate. One mechanism is local cell-to-cell interactions that are mediated via surface receptors. And this is where inductive signaling comes in. The interactions of one cell with those that are nearby are very important in influencing the fate of that cell as differentiation progresses. So we can think of these local interactions as a neighborhood effect, that the neighborhood of the cell will influence the ultimate fate as inductive signaling takes place and begins to specify the final destiny of an individual precursor cell. So the second major factor that can shape the ultimate destiny of a neuroblast is cell lineage. That is, what is the transcriptional history of the cell, and what was the parent cell that gave rise to that particular progeny. So together, these cell-to-cell interactions mediated via surface receptors and inductive signals, together with transcriptional history, influence the ultimate destiny of a given neuroblast and its final location, its final form, and ultimately its functional position within the networks of the developing brain. Well, there's one final factor that I want to speak about that is very important in brain development, and perhaps it's a surprising factor that you might not have necessarily thought was important in brain development, and that factor is the death of brain cells. This is a process that we call apoptosis, and this refers to programmed cell death. Indeed, proliferation of neuroblasts produces in many regions of the brain roughly twice as many brain cells that ultimately survive. And it's through a process of programmed cell death that we achieve the final complement of cells in the brain that we then carry into postnatal life and have with us through the remaining years of our life. So apoptosis becomes an important mechanism for establishing the appropriate complement of cells in the different gray matter structures in the human central nervous system. Well, this process of cell death reflects the expression of what some have called suicide genes. These are genes whose products induce the dissolution of cellular constituents that ultimately lead to the death of the cell. I'm not a particular fan of this Darwinian perspective of brain development. I think it's not so clear in what sense are cells that die less fit than those that don't express this programmed cell death pattern. Rather, I think it's more likely that there are particular factors that are acquired that support the survival of neurons, and a failure to acquire those factors may lead to the induction of the suicide genes. So the cell itself may not be less fit in any particular way, it just may not be privileged in a means of acquiring a particular trophic factor that is necessary for survival. We'll say more about trophic factors in a later tutorial. But for now, I just wanted to impress upon you the importance of cell death. So this implies that there might actually be some optimal size to the human brain with some particular complement of brain cells. Well, by optimum, I mean that one can have a pathologically small brain, but perhaps one can also have a pathologically large brain where there are too many neurons, insufficient programmed cell death. We think that's likely, but the range in the middle is actually quite enormous when we consider the variation in brain size among human individuals. And even within particular regions of the brain, there can be as much as a twofold difference in the size of particular cortical areas, such as the motor cortex or the visual cortex or the prefrontal cortex. These are regions where this question has been studied. Presumably, this variation in size reflects variation in either the proliferation of brain cells or the programmed cell death of those cells in early brain development. What we still don't know is what is the functional consequence of this variation in brain size. I've thrown this out to you. Is bigger better? I think that in terms of overall brain size, we certainly don't have any principled way to answer that question affirmatively. There is good reason to argue that perhaps better is better when it comes to human brain size. That is, better might reflect more efficient function, not necessarily more processing units. So some of this variation in size undoubtedly reflects a differential complement of brain cells that have come to populate specific regions of the cerebral cortex. And it may be that on a local level, that the size of the circuit, the complement of brain cells that contribute to a given neural circuit might be important for functional capacity. This remains an open question and one that's subject to much ongoing research and considerable debate, I'm sure. Well, what I've tried to convey in this tutorial is a bit of the story as to how do brain cells get to their final destination in the developing central nervous system, and how do they differentiate into their final fate, be they a sensory neuron, a motor neuron, a glial cell, a neuron in the upper layers of cortex that are computing new properties as incoming information from the thalamus is relayed up to those upper cortical layers, or perhaps there are lower cortical neurons that send axons back down to subcortical structures like the thalamus, the basal ganglia, even the spinal cord in the case of the motor cortex. Well, the next phase of the story of brain development is indeed the formation of neural circuits. So I want to speak to you next about the outgrowth of neuritic processes that form dendrites and axons and ultimately the synaptic connections among neurons. So the story continues, and I'll see you next time as we talk about the construction of circuits in the central nervous system. Welcome back to this next tutorial in our series on the changing brain. Today we're going to talk about the construction of neural circuits. And this topic will pertain to three of our core concepts in the field of neuroscience. Again, we're going to be talking about a different kind of neuronal communication. Communication that is mediated chemically, but in this session we're going to begin to introduce the concept of building synaptic connections, and that will enable both chemical and electrical communication to be possible in developing neural circuits. So we're going to see the genesis of this very important core concept in the field of neuroscience today as we consider the construction of circuitry. We're going to talk about circuits that are in large measure genetically determined, circuits that provide the foundation for behavior, such as the ability to detect potentially painful and damaging stimuli in our skin surfaces. And then lastly, we'll set the stage for a more rich discussion that I hope to have with you in our next tutorial about how life experiences can modify the structure and function of the central nervous system. And I'd like to introduce a hypothesis, really, that will allow us to understand how can chemical signals that have the ability to change the course of neuronal development be modulated by activity. And this hypothesis will then help us understand how life experience can change the structure and function of the brain. Okay, well, let's consider the learning objectives that I have for you in this tutorial. I want you to be able to understand what is a principal organelle that is responsible for pathfinding in developing axons, as well as the growth of dendritic arbors. And this structure is called a growth cone, so I want you to be able to characterize the structure and function of growth cones. I want you to be able to discuss the major classes of signaling molecules that guide axonal growth in the developing brain. And I want you to be able to have a cogent discussion of neurotrophins, and I want you to be able to discuss the mechanisms of neurotrophin signaling. Well, hopefully all of these topics will make sense to you after spending a little time with me in this tutorial. So let's jump right into it. Well, I would remind you of our broad framework for understanding the forces that influence the developing brain. We talked quite a bit in our last tutorial about the concept of nature in brain development. That is, the lineage-derived signals that result from the expression of particular genes. And so this influence in brain development can be considered genetic specification. Well, today we're going to talk about the construction of neural circuits, which begins to engage our two other major sets of factors that influence the final shape and structure and function of neural circuits. Well, if indeed there is ever a final shape to them. What I have in mind is self-organization. Self-organization is mediated via activity-based mechanisms that play out as cells acquire the capacity to generate electrical signals. And that begins to shape the expression of a variety of phenotypes that circuits begin to acquire as they build out their connections. Well, as the developing embryo, fetus, and into postnatal life, developing infant begins to interact with the environment, then sensory motor experience becomes an important shaper of circuitry in an ongoing phase of circuit construction that continues into postnatal life. So the story of the construction of neural circuits begins in utero. It begins at a relatively later stage in embryonic development in the third trimester, but it will continue well into postnatal life. So many of the mechanisms we'll be talking about today reflect nature, that is genetic specification. However, we will begin to describe some of those factors that play an important role in mediating the influence of nurture. And again, that's going to be more primarily the topic of the next tutorial in this series. Okay, well, with that as a broad introduction, let's talk about some of these mechanisms that pertain to genetic specification and self-organization in early brain development. Mechanisms that will shape the growth and the viability of neurons and their connections in the developing central nervous system. Well, the first set of mechanisms that play out establish polarity in the developing neuroblast. Now, you'll recall that the neuroblast is derived from an epithelium, and all epithelial cells acquire a polarity. That is, there's an apical surface and a basal surface to their cells. And this process plays out in developing neuroblasts, although their apical and basal surfaces are increasingly difficult to recognize because increasingly these cells no longer resemble the columnar epithelium that first established the neuroectoderm. Nevertheless, there is the establishment of polarity such that from the apical surface of the developing neuroblast, an axon will emerge. And from a basal surface, dendritic branches will begin to grow out. Now, initially, with the outgrowth of these processes, we really can't tell whether they are destined to become axon or dendrite. We simply call them neurites. But as development progresses, a single axon becomes recognizable and differentiated from the apical aspect of the neuroblast. And that axon begins to acquire a distinctive organelle at the growing tip of that axon. That's the structure that we call the growth cone. And let's spend some time together thinking about the growth cone and what makes it such an important structure in the finding of axonal pathways in the developing brain. Well, if one were to look at the tip of an axon that is emerging, let's say, from a cell body somewhere in this direction, one would find that there is a large flattened structure that begins to develop as that axon begins to differentiate from just a neurite that might be extending away from this neuroblast. So this structure is what we call the growth cone, and it's characterized by a fairly flat laminar type of appearance. We call this a lamellipodium or a sheet foot. That's one way to understand that term. And then from that lamellipodium extend these long finger-like processes called filopodia. Now what we're seeing in this figure is an illustration zooming in just on one portion of the growth cone, looking at a filopodium. And this allows us to understand just a little bit about the dynamics that shape this growth cone as it interacts with the environment of the developing embryo. So what we find in this lamellipodium is a rich supply of mitochondria to supply the energy that's necessary for the activity of the growth cone. We also find a variety of filaments. We find actin filaments that assemble in the filopodium, and this allows that filopodium to have a highly dynamic behavior as actin polymers are assembled from the monomeric elements. So the assembly of those filaments is responsible for the protrusion of a filopodium, and then the disassembly of those filaments allows for the retraction of a filopodium. Meanwhile, microtubules are being assembled and disassembled in the lamellipodium. So all of the machinery that's necessary for the construction and deconstruction of these filaments needs to be synthesized and then localized to this growth cone. Now, these processes are responding to signals in the environment through which this growth cone is exploring. And those signals are transduced via a variety of receptors that are present within the leading edge of this growth cone. So, for example, one can find a variety of channels, including our transient receptor potential channels that can mediate the interaction of this growth cone with chemical signals in the environment. There are other means by which calcium can enter this lamellipodium. And as you know, calcium is a very important second messenger system within all cells, and in particular within developing neurons. So calcium becomes a key mediator of the events that are detected at the plasma membrane of the growth cone, which then are transduced into changes in the structure and growth patterns of this growth cone. So this entire growth cone is a highly modal structure. So this growth cone can explore the environment within which this axon might be proceeding. It can decide that it needs to change directions, perhaps retract from growth in one direction and begin to extend growth in a different direction, perhaps an orthogonal direction to the original pattern of growth. So there is a tremendous amount of dynamism associated with this growth cone as it actively explores the environment, responding to chemical signals. And sometimes there is an important point of decision, is there not? There are what we might call forks in the road for a developing axon that's pioneering a pathway. We think of these forks in the road in particular at the midline of the developing embryo, where a variety of axonal projections must make a choice to cross the midline or not to cross the midline. Well, I thought this might be a helpful point to insert a study question to challenge you to review some of what you know about anatomical pathways, mainly those that you learned in Unit 3 of the course. So let's just pause for a moment here and take on the following study question. Hopefully you were able to come up with what I was looking for in that study question. If not, you'll see it appear shortly as we progress in this tutorial. All right, well, let's consider now what are some of those molecular signals that guide the growth of the axon, that influence the decisions made by that growth cone as it explores the environment through which that elongating axon is traversing. Well, we consider basically two classes of molecules. There are molecules that are associated with membranes of cells of the developing embryo. And these molecules may also be embedded in the matrix that lies between the cells. We call that the extracellular matrix. So these are bound molecules, cell-associated molecules, or extracellular matrix-associated molecules. That's one category. Another category are diffusible molecules. So as that implies, these are not cell-associated, they're not bound to a membrane or to the matrix, but rather they are freely diffusing in the extracellular spaces of the developing nervous system or the developing embryo. So let's look at specifically what we have in mind here. So again, these cell-associated molecules are on the surface of cells, or they're embedded in the extracellular matrix, and they mediate molecular interactions between the developing growth cone and the parenchyma, or that is whatever the substratum is that that growth cone is exploring. Now, the interactions between the growth cone and the surfaces can trigger second messenger systems within the growth cones that transduce extracellular signals into patterns of directed growth. Some of these cell-associated molecules play a role in stabilizing the linkages between axonal membranes and the structures that surround them. One important element will be the glial cells that begin to form myelin or begin to lay down a substratum over which that axon will be laid down. So there needs to be not just direction of growth, but also stabilization of the axon that is elongated in the wake of that growth cone. Now, the diffusible molecules, as I mentioned, they are secreted by target structures and diffuse into the extracellular spaces. So then by definition, the diffusible molecules can act over a much greater distance within the central nervous system or outside the CNS in the embryo than can the cell-associated molecules that are anchored to a membrane or the extracellular matrix. Okay, what I'd like to do next is to consider some specific examples of our cell-associated molecules to give you a feel for what kinds of actions they may mediate. So here's a figure from your text that just illustrates some of the important cell-associated molecules that we know about, beginning with those that are associated with the extracellular matrix. So here is a representation of that matrix, and the idea is that there are proteins and other kinds of molecules that present sort of a fibrous matrix in the extracellular spaces. And molecules can interact then with receptors, which are often the very same molecule. We call this a homophilic interaction because it's the same molecule interacting with itself. In this case, what we see are interactions among the integrin receptors. Now, these integrins themselves are not do not have enzymatic activity in the cytoplasmic surface of the molecule, but rather they interact with other kinds of molecules that do have that enzymatic activity. For example, kinases and other kinds of signaling molecules that can activate other second messenger systems for further signal transduction downstream. Well, those are the integrins. There's a variety of individual members of that family. Next, I'd like to mention the cell adhesion molecules. These are very important for mediating several different specific aspects of pathway development. We'll talk about some as we move along here. There are certain classes of cell adhesion molecules that don't require the activity or the presence of calcium. And so that's what's shown here in panel B. So these cell adhesion molecules, or CAMs for short, that are depicted here would be the L1 CAM, as well as the neuronal cell adhesion molecule called NCAM. So these do interact with similar kinds of kinases and other effector systems within the cell that can influence transduction cascades within the cell that's interacting with these molecules. Now notice again, there's homophilic interaction of one kind of cell adhesion molecule with the very same type of molecule on the surface of another cell. Now there are cell adhesion molecules that are calcium dependent. They require calcium for their activity. Here's an example of one. It's called a cadhedron. So here we have cadhedrons that require the binding of calcium ions in order to engage their activity. And these activities then are mediated via a variety of second messenger systems, some of which can have positive effects, some can have negative effects depending upon the system in question. This particular cadhedron interacts with the beta-catenin pathway, which can have an impact on the assembly of actin filaments in the cell that is receiving this signal. And then lastly, I would mention the ephrins. So the ephrins are a very important class of molecules. The ephrin proteins are presented on the surface of cells, and they interact with receptors that we call Ephs, or ephrin receptors. These are tyrosine kinase receptors, and they can activate a variety of different kinds of second messenger systems within cells. Here we see an ephrin receptor interacting with the Rho GAP system that likewise can have an impact on the assembly or disassembly of actin filaments. Now I'll give you one example of the importance of ephrins and ephrin receptors in the development of circuitry. Let's consider, for example, the development of a topographical map. This requires axons from one kind of structure in the developing nervous system to project into a target structure, and to do so in an orderly way. We saw many examples of this as we talked through Unit 3 and considered our sensory systems, and perhaps we took it for granted that it is possible to establish such topographic maps connecting one station in a neural pathway with another. But it's really quite an amazing accomplishment in neural development. And for a long time, neuroscientists suspected that there may be chemical molecules that somehow are establishing the basic framework for a map, and then in subsequent stages of development, perhaps neural activity can be used to refine those connections. And indeed, that seems to be the basic paradigm by which topographic maps are established. We now know that ephrins and ephrin receptors play an important role in establishing topography in such systems. And here's an example that demonstrated rather dramatically the nature of the recognition between one supply structure and its target in the brain. And here what I'd like to present is a bit of a scenario derived from studies of an amphibian system, the frog visual pathway, which connects the retina to the frog's version of the superior colliculus that we call the optic tectum. And what's known about the anatomy of this system is that the posterior or the temporal part of the retina from the frog eye tends to send its axons into the anterior region of the tectum, preferentially avoiding the posterior region. Meanwhile, the more anterior or nasal part of the frog's retina sends its axons past the anterior part of the tectum to arborize mainly in the posterior part. Well, one fairly dramatic demonstration that there must be some kind of recognition that allows temporal retina to affiliate with the anterior part of the tectum and nasal retina to affiliate with the posterior part came from experiments that involved rotating the eye of a frog. Now, unlike mammals and many amphibians, if you destroy the optic nerve, it will actually regrow into the brain. And what was done in this experiment is that the eye was surgically rotated and then reattached. And after some time, the axons that were cut in the optic nerve were able to regenerate and reinnervate the brain. And so the behavior of the frog was then observed, and what was very clear is that after rotating the eye of the frog, its behavior was precisely off by about 180 degrees, which was the extent of the rotation. So a visual motor target appears to the upper left, and the frog protrudes its tongue to the lower right. Not good for the frog, maybe good for the fly. It indicates the identity of the retinal ganglion cells continued to be recognized by the appropriate domain of the optic tectum, irrespective of the surgical rotation of the eye. So what this experiment illustrated is that the anatomical structures of the eye remapped to the same divisions of the optic tectum, irrespective of the rotation of the eye. So in this case, we imagine that the fly is out in the temporal part of the visual field, which ought to be seen with a normal eye by the nasal part of the retina. The problem is that the eye has been rotated. So now what should be the temporal part of the visual field is being seen by the temporal part of the retina following rotation. Well, that part of the retina is going to innervate and project into this anterior part of the tectum, which is going to be responsible for inducing this reflexive motor action down into the right, which is now exactly 180 degrees off of where the appropriate strike should have been to acquire the target. So this experiment shows us that this mapping seems to be of a chemical nature rather than a functional nature, fully tuned and fully accommodated to the behavior of the animal. So many years had passed, decades as a matter of fact, that established this topographic map were worked out. So we know that in this system, what mediates the topography of this connection between the retina and the tectum are efferents and their receptors. So rather than there being a precise lock and key mechanism on a point-by-point basis, what we see is the establishment of gradients. Gradients of the ligand and gradients of the receptors. And the matching of ligand concentration to available receptor is what establishes the continuity in the topographic mapping, such that the most posterior temporal portion of the retina is mapped to the most anterior portion of the tectum. And then a gradient is established for the rest of the posterior temporal retina. And that gradient then is what allows for the exclusion of axons that then come to be mapped into the posterior part of the tectum, those axons being derived from the nasal part of the retina. So efferent signal is critical, we think, for the establishment of topographic maps, not just in the optic tectum of the frog, but we think wherever we find topographic maps in the developing central nervous system, including ours. Okay, so I introduced two categories of molecules that help mediate the signaling that's required to grow circuitry in the developing nervous system. We've talked about cell-associated molecules. Now let's talk about the diffusible molecules. And they're essentially molecules that are secreted by target tissues and diffuse out into the extracellular fluids. These molecules can affect the growth and the survival of neurons, and their individual processes, whether they be dendritic branches or axons. There are two categories of diffusible molecules based on their mechanisms of action and how they affect the development of the neuron and its processes. I'd like to talk first about tropic molecules. These are molecules that guide the growth of axons toward or away from the source of this particular molecule. So the directed growth is what we mean when we talk about tropism, and so this is a tropic molecule that directs the growth of an axon. They come essentially in two functional valences. There can be chemoattractant molecules or chemorepellent molecules. So I'd like to give you some examples of each. There are also trophic molecules. So while tropism refers to directed growth, trophism refers to nourishment. So these are molecules that provide some form of nourishment. Now, not nourishment in the form of glucose to be burnt up or ATP to be hydrolyzed and release energy from the chemical bonds, but nourishment in a way that promotes the survival and growth of the neurons and their processes. In the peripheral nervous system, these trophic molecules become really important in matching the axonal innervation with the target tissue that's present. And we'll also see the importance of trophic molecules for the development of appropriate circuits within the central nervous system as well. Now, one very important and very interesting aspect of trophic molecule production is that this is activity dependent. So this will provide the basis for competitive interactions in developing circuits. So a very interesting concept. We'll say more about that in our next tutorial. So let's consider some examples of our diffusible molecules. So again, the general mode of operation here is that these are molecules that are secreted by target tissues, and these molecules interact with receptors that are present on the surface of the growth cone. These diffusible molecules interacting with their receptors can have widespread effects on the neuron that transduces these signals. There can be local effects that can affect the growth patterns at the level of the growth cone and the individual axonal branch in question, but there might also be signals that go back to the cell body and affect gene transcription in the entire cell. So there are a variety of consequences of the interaction of these diffusible molecules with their receptors. So in this figure, we see illustration of several different kinds of diffusible molecules and their effect. We see some source of diffusible chemoattractant signals here that begin to divert the growth of an axon towards that source of that chemoattractant. We also see contact-mediated chemoattractant signals that are important for helping to establish the growth of axons in parallel, and this is critical for the formation of axonal pathways where there are many hundreds of thousands of axons typically growing together in parallel. Now there are also chemorepellent signals that can be produced at some local source. And you'll see that as a growth cone begins to approach and explore, it may interact with one of these signals and then be diverted away to grow in the opposite direction from that source. There may also be contact-mediated chemorepulsant signals that help to make sure that the growth cones at the leading edge of the developing pathway begin to explore the entire environment, giving these growth cones an opportunity to seek out appropriate sources of chemoattractant and avoid the appropriate sources of chemorepellents. Now also in this figure, we see the presence of trophic molecules that are expressed, and they don't necessarily direct the growth of an axon into the source of that trophic support. Rather, what they do is they validate that growth. So the growth is directed by the tropic molecules, but the survival and the ability of that axon in the neuron that gave rise to that axon to thrive is dependent in some measure on the acquisition of trophic support that comes through the interaction of these neurotrophin molecules and their receptors. We'll say more about that in just a moment. First, I'd like to give you some examples of some of the tropic molecules and then talk about one very important pathway that illustrates the operation of these molecules in development. So there are several representatives of these tropic molecules that we now know about. On the left, we see examples of the netrin and SLIT family of tropic molecules. Netrin is a soluble protein that is released and interacts with its receptors, DCC, and an associated protein called UNC5. So these interactions can result in the assembly of actin filaments, leading to the directed growth of that growth cone and its axon being elongated in its wake towards the source of netrin. Now there's a related tropic molecule called SLIT that interacts with a receptor called ROBO, and SLIT antagonizes the activity of netrin. So netrin and SLIT together provide sort of a push-pull system or a go-stop kind of system that allows for the direction of growth, and this is particularly important at those forks in the road, as I'll show you in just a moment. Now, another diffusible tropic molecule illustrated here are the semaphorins. So semaphorins are cell-associated molecules that can be released and diffuse away from their source, and the semaphorins can interact with receptors, two of them illustrated here, plexin and neuropilin. These receptors can likewise interact with transduction machinery that can affect the assembly of cytoskeleton and the patterned growth of that growth cone. However, what the semaphorins tend to do is promote the disassembly of these neurofilaments, and that results in the retraction or the collapse of the filopodia and the lamellipodia. So the semaphorins are important signals that mediate the repulsive signals that are encountered as axons explore their environment. So let's consider one pathway, and indeed this is the pathway that I had in mind in our study question a little while ago. This pathway is the spinal thalamic tract. Now recall the second order axon of the spinal thalamic pathway. It has to do something which I find completely remarkable. This axon is grown out by a neuron in the dorsal horn of the spinal cord. And this happens throughout the entire length of the spinal cord. The axon will grow out very near its level of origin, and it will begin to grow in the anterior medial direction. So that axon encounters the midline of the spinal cord in the ventral white commissure, the anterior white commissure of the spinal cord. And it makes an important growth choice. It grows across that midline and enters the anterolateral white matter on the contralateral side of the spinal cord. So there is a decision to cross the midline here in the outgrowth of this second order axon. But there's also one more remarkable aspect to the growth pattern. Once across the midline and into the anterolateral part of the white matter, this axon now makes a hard turn in the superior direction and grows to the ventral posterior complex of the thalamus. So the implication is that there must be some chemoattractant signal that draws this axon towards the anterior midline of the spinal cord. And then once across the midline, there must be some signal that directs the growth away from that segment of origin and towards the superior axis. So we imagine there may be some combination of chemoattractant and chemorepellent molecules that achieve the necessary pattern of growth for this second order axon to establish the spinal thalamic tract. And indeed, what we imagine happening with the growth of this pathway is these second order axons, these commissural axons, are attracted towards the region of the floor plate. And they're attracted by the presence of netrin, which is secreted by this special region of the neuroepithelium called the floor plate. So these axons begin to grow in towards that floor plate. And then once they grow across the floor plate, now slit seems to be produced just lateral to the location of that floor plate. And slit now begins to negate the influence of netrin. And there's likely some other kind of signal, some other repulsive signal that is present here in the anterolateral part of the developing neural tube, perhaps a semaphorin, which will collapse the growth cone from growing in this direction and rather promote growth away from the source of that chemorepellent. There very likely is also some kind of chemoattractant that is in some concentration gradient drawing this axon towards the superior aspect of the developing hindbrain, even into the forebrain, to reach its ultimate target in the diencephalon. So this is a wonderful example of how the presence of chemoattractant and chemorepellent molecules can mediate what are really some spectacular patterns of growth that we've learned about in our survey of the structure of the sensory and motor systems. I'll refer you to a beautiful box in chapter 23 that talks about another famous fork in the road. That would be the optic chiasm, where similar functions seem to direct the nasal retinal axons across the optic chiasm while repelling the temporal retinal axons, which remain on the ipsilateral side of the brain, progressing through the ipsilateral optic tract. So turn to the box there in chapter 23 to learn more about that particular pathway. Now, I'd like to turn to a discussion of the trophic molecules and consider the nature by which these molecules provide nourishment that promotes the growth and survival of neurons and their processes. And I'd like to take you through a really classic experiment in neuroembryology done in the first half of the 20th century by two real heroes of mine, Victor Hamburger, who did most of his work at Washington University in St. Louis, where I did my own graduate studies. I had a chance to meet Victor Hamburger towards the latter years of his life, and it was a real joy to do so. Victor Hamburger is a role model for so many reasons. He was a wonderful scientist, but he had a particular perspective on collegiality, which has certainly enriched his legacy for those of us that know something about him. Victor Hamburger began to be aware of a Italian scientist, a young woman who was working under extraordinary conditions. Her name was Rita Levi-Montalcini, and Hamburger and Levi-Montalcini, a world apart, were doing very similar experiments and not always coming up with agreeable findings. And rather than developing a sort of a competitive adversarial relationship, Hamburger invited Levi-Montalcini to join him in his group in St. Louis, which she did after working through tremendous hardship in the time of World War II. Well, eventually Levi-Montalcini and Hamburger, together with Stanley Cohen, collaborated in a landmark series of experiments that led to the discovery of neurotrophins and a deeper understanding of how they impact the development of the nervous system. Well, here's some simple experiments that they performed. It involves a study of the development of motor neurons in the spinal cord relative to the muscles that they innervate. Now, as I mentioned in our last tutorial, many more neurons are typically produced in each region of the nervous system than actually survive, and that's true here in the spinal cord. What we see in the ventral horn in this illustration in red are those neurons that actually go on to survive the course of development. In green is a population of neurons that are initially produced but die off through a process of programmed cell death. So why should this be? This was a question that Hamburger wanted to explore. And what was the experiment here was to take a chick embryo and delete one of the limb buds, which would have obviously a dramatic impact on the amount of target tissue that would be available for the outgrowth and connectivity of the alpha motor neurons on that side of the appropriate enlargement of the spinal cord. So the result of this unilateral limb blood ablation is that on the ipsilateral side of the spinal cord, there's a dramatic reduction in the population of motor neurons, whereas on the intact side of the spinal cord, the normal complement of motor neurons survives the process of development. So this suggests that there's something about the presence of target tissue that's essential for the survival of the neurons that normally would come to innervate that tissue. Well, in the chick embryo, it's possible to do the complementary experiment. So what Homburger and his colleagues did was they took a limb bud from one embryo and transplanted it to another, essentially doubling the complement of target tissue that would be available for the innervation of motor neurons from the appropriate segments of the spinal cord. And then after about a week of growth, what was discovered is that on the side of the embryo that has the extra limb bud, there are roughly twice as many neurons that would normally be present. And the control side is the index that allows a conclusion regarding the impact on cell survival from the presence of that extra limb bud. Now, one might have imagined that these extra neurons were somehow induced through mitotic activity here in the ventral horn following this addition of this extra limb bud, but through a variety of experiments we know that that's not the case. The extra neurons that are present here reflect the survival of neurons that otherwise would have died through programmed cell death. So this suggests that there's some factor that's present and released by target tissue that is essential for the survival of neurons, and that such factors might be important in establishing the appropriate complement of neurons that connect to target tissues. So these experiments allow for two principal conclusions. That there is decreased programmed cell death of a motor neuronal pool with augmentation of target tissue, that is, with the presence of an extra limb bud. But it also illustrates that with reduction of target tissue, there's an enhancement of programmed cell death, leading to a depletion of those neurons that otherwise would have survived. So this suggests that target tissues release some kind of a substance. We now call that substance a trophic factor, that is essential for the survival of neurons. And this substance might actually be quite critical for establishing the appropriate complement of neurons that innervate a target tissue. Now, we think that through a variety of other experimental evidences, we believe that trophic molecules also operate at the level of individual axonal branches, not just the survival of the entire neuron. And this becomes critical for establishing the appropriate patterns of convergence and divergence as neurons innervate their target tissues, be they targets in the periphery or targets within the central nervous system. So here's two examples from the peripheral nervous system. In early development, alpha motor neurons innervate individual muscle fibers, and it's possible to find multiple collaterals from different alpha motor neurons supplying the very same muscle fibers. So here in this illustration, for example, a single alpha motor neuron might send branches that innervate each of three individual muscle fibers. In autonomic ganglia, we may see something quite similar. There may be collateral branches from a single preganglionic neuron that innervates multiple postganglionic neurons. Now, this is quite intriguing because we know that in the mature nervous system, a single muscle fiber is supplied by a collateral from just one alpha motor neuron. And likewise, a ganglionic neuron that lacks dendrites, such as what we have illustrated here, receives its synaptic inputs from a single preganglionic neuron. So there's some kind of sorting process that must play out here to establish this appropriate degree of convergence and divergence. And a key mediator of this competition seems to be neurotrophins. Well, a variety of studies done in these systems and others have given rise to a set of propositions that we sometimes call the neurotrophin hypothesis. So the neurotrophin hypothesis goes as follows. We imagine that in development, the survival of neurons and their axonal connections depends upon the availability of a trophic factor that's present in some minimal quantity. Such trophic factors are synthesized and released by target tissues. And again, we think that they're present only in very limited quantities. And this sets up a competitive scenario where the survival of neurons and their specific connections depends upon successful competition for the acquisition of that trophic substance that's present only in limited supply. And in this way, we imagine that for that single muscle fiber, only one axon can acquire sufficient trophic substance to promote the survival of that connection. Similarly, for a ganglionic neuron, so that only one preganglionic neuron can acquire sufficient trophic support in order to maintain the viability of that connection. Now, let me just back up for a moment and highlight an interesting and accurate feature of this illustration. Notice that there are fewer connections in these immature neurons compared to the more mature form. So the overall numbers of synapses is increasing even as competition is playing out, and the numbers of inputs, that is the number of individual axon fibers or axon collaterals that come to innervate a given postsynaptic cell, is decreasing. So there is a winnowing down of the numbers of neurons that are interconnected, even while there is a net increase in the numbers of synapses that are being formed on these postsynaptic targets. So in this autonomic ganglion, there may be a winner-take-all outcome here, such that all of the postsynaptic targets that are available on this ganglionic neuron are now occupied by synapses that are elaborated by a single preganglionic axon. Okay, let's get back to our neurotrophin hypothesis. So we've emphasized that in brain development, this is going to be really critical for mediating competitive interactions among different axons that may be competing for the same postsynaptic targets. We'll see some examples of that in the developing visual system in our next tutorial. But I'd like to just make a point, and that is that in the mature nervous system, neurotrophins remain very important molecules in shaping the structure of circuits in the central nervous system. And this is especially true in the context of plasticity following injury. One phenomenon that we'll explore later in this unit is that as nervous tissue is recovering from injury, many of the mechanisms that were important in establishing connections in brain development become reactivated. So many of the genes begin to express the substances that were important in building connections in the first place as a means for promoting regeneration and regrowth of connections within the central nervous system. Now, this process is modulated in various ways. We'll eventually come to that. But for now, I want to emphasize that neurotrophins remain important in shaping the structure of connections even in the mature brain. And this is especially important in considering how the brain responds to injury and disease. But it's also important in understanding how sensory motor experience can shape connections within the brain and thereby instill learning. So, for example, when we acquire a new motor skill, when we practice a sport or a musical instrument or a new dance step, we imagine that there is some kind of plasticity that's taking place. And neurotrophins very well may play an important role in mediating the competitive interactions that we hope will eventually build a more efficient circuit that will reinforce those patterns of connections that instantiate the acquired skill. So let's talk a little bit more about the neurotrophins and their receptors. So we know that there is a family of neurotrophins now. The most famous and first discovered neurotrophin was nerve growth factor, NGF, but we've since identified at least 3 other neurotrophins. There is an important molecule called brain-derived neurotrophic factor, BDNF. There are other neurotrophins that we call NT45 and NT3. So these all bear some structural similarity, and their mechanisms of actions also are similar. The neurotrophins interact with a set of receptors that have tyrosine kinase activity on the cytoplasmic surface. So we call these TRK, or TRAC receptors for short. So the TRAC-A receptor interacts with nerve growth factor. The TRAC-B receptor interacts with BDNF and NT45. And the TRAC-C receptor interacts with neurotrophin 3. Now, what are the consequences of receptor activation? Well, the consequences are incredibly diverse. And as is usual for receptors that engage metabolic processes, the impacts of receptor interaction with its ligand can be as diverse as the second messenger systems that are present in that neuron. And this is the case for our TRAC receptors. There are TRAC receptors that interact with various kinds of signaling pathways. I won't go through the details of each, but I'll just make the point that the distinctions among these second messenger pathways are what accounts for the differential effects of neurotrophins. Some neurotrophins will affect specifically cell survival. Others will work through a variety of pathways that mediate activity-dependent plasticity and the capacity to respond to coordinated patterns of electrical activity. Other neurotrophin-mediated pathways will have a more restricted effect on the survival of individual neurites, that is, individual axon collaterals or dendritic branches. Or some of these pathways might influence the transcription of genes that allow for the further differentiation of the neuron. Well, these neurotrophins and their receptors are a picture of the signaling that takes place after these neurotrophins have been processed and differentiated through post-translational modification. But before that happens, these neurotrophins interact with another receptor that we call the P75 receptor. So the P75 binds to the relatively unprocessed forms of these neurotrophins that are synthesized and released. The processing happens after they're released and as they interact with enzymes and proteases that are present in the extracellular spaces. This P75 receptor interacts with the relatively unprocessed form of the neurotrophins, which is what we find when they are newly released into the extracellular spaces. Thereafter, they can be processed and cleaved in various ways, modified by enzymes and proteases that are either bound to the surface of membranes or present in the extracellular matrix. The consequences of P75 activation likewise can be diverse. One can impact the growth of axons and dendrites. One can impact either promoting the survival of the cell or activating the genes that result in the death of the cell through the process of apoptosis. So again, diversity of second messenger systems as a consequence of the interaction of the neurotrophin and its receptor. Now, some of these neurotrophin effects are actually taking place in the cytoplasm and even in the nucleus of the neuron. So there must be some means by which the signal can process in the retrograde direction from the growth cone or the developing synapse back to the cell body. So how does that happen? Well, we now have a increasingly detailed understanding of how the neurotrophin and its receptor can become internalized and then associated with the transport machinery that allows that internalized structure to be shipped back to the cell body, where the neurotrophin and its receptor can mediate additional impacts on the growth and differentiation of the neuron. Well, this can happen as neurotrophin interacts with its receptors, and this complex then engages a set of scaffolding proteins that can internalize this receptor complex, forming an endosome that can then affiliate with the molecular motor along a microtubule network that allows this complex to be translocated back to the cell body. And once in the cell body, then the receptor can engage other kinds of second messenger systems that may be present there that can impact the growth and the survival of that entire neuron. And finally today, I'd like to speak to you about the molecular mechanisms that are beginning to be understood that are responsible for the formation of synaptic connections. Well, this begins when a presumptive presynaptic element is recognized by some postsynaptic target site. And there are calcium-dependent adhesion molecules, or cadherins and protocadherins, that seem to be responsible for this early recognition event. And following the interaction of these adhesion molecules, the early differentiation of the presynaptic and postsynaptic elements begins to take place. So it's very likely, as a result of activation of these adhesion molecules, that we begin to see the formation of vesicles and the machinery that will become essential for processing these vesicles in the presynaptic terminal. Well, as the process ensues and additional specialization begins to be induced at that presynaptic site, we know that a number of other factors become very important. And we begin to see the assembly of a network of inductive signals that begin to influence the construction of the presynaptic side of the synapse, which involves the formation of an active zone and all the metabolic machinery that hopefully you will recall from our studies of neural signaling in Unit 2, as well as the construction of the postsynaptic side of the synapse, which involves the assembly of ion channels and second messenger systems and the organization of a postsynaptic scaffold that will allow for the trafficking of receptors to that postsynaptic membrane and away from it. Well, we know something now about some of the molecules that are involved. There are additional families of cell adhesion molecules that are important. Efferent signals become important. And one very important substance that seems to have an early impact on this stage of maturation is called neuroregulin. Well, all of these factors are operating here at this complex of proteins, and let's take a closer look at that. So here is neuroregulin, which is a presynaptic protein that can be released and can diffuse into that presumptive postsynaptic cleft, where it can begin to serve as an inductive signal, which begins to assemble all of these proteins that are important for setting up that postsynaptic site. Eventually, two other molecules become very much involved, one called neurexin on the presynaptic side and neuroligin on the postsynaptic side. On the presynaptic side, neurexin is important for beginning to build up this active zone where synaptic vesicles begin to interact with snare proteins and other important proteins, including synaptotagmin, which is that molecule that detects the presence of presynaptic calcium. One also needs to cluster voltage-gated calcium channels near this active zone, such that calcium influx can be local and it can be rapid leading to the development of a fusion pore and the exocytosis of the contents of these synaptic vesicles. Well, all of this machinery needs to be aggregated and organized in a coherent way for the formation of a viable presynaptic element. Neurexin seems to be an important signal that induces this organization. On the postsynaptic side, neuroligin does complementary functions, aggregating the protein complexes that are essential for the insertion of appropriate receptors, such as our AMPA receptors for glutamate or our NMDA receptors, as well as providing for the aggregation of postsynaptic density proteins that are critical for the proper trafficking of these receptors up to that postsynaptic membrane or their withdrawal from that membrane. In addition, there are second messenger systems that become affiliated with various types of receptors, as you now know, and all of this has to be orchestrated in a way that allows for the postsynaptic cell to respond to the chemical message released by the presynaptic element. So this is a progressive series of events that are mediated via inductive signals that operate over a time course that establishes the structure and function of the mature synaptic connection. So before we conclude this tutorial, I'll leave you with one final study question that will hopefully focus your attention on neurotrophins and the important role they play in mediating the establishment of proper connections in the developing nervous system. So I'll see you next time, and I'll have the privilege of sharing with you some of the research work that I've been involved with together with my collaborators over the years. And I'll have a chance to talk to you about one of my favorite topics, which is the influence of experience in shaping the structure of neural circuits in the developing brain. I'll see you then. Thanks for listening, and hope you enjoy your studies of neural development. Welcome back to Medical Neuroscience, and welcome to this tutorial on the modification of neural circuits in early neonatal life. Our core concepts that pertain to this session are, first of all, that life experience affects the function and the structure of the nervous system. This is really the whole point of this discussion today, is to talk about how sensory motor experience changes the nervous system. Well, for me anyway, this topic certainly pertains to one of our core concepts in neuroscience, and that is that our human brain is what endows us with a natural curiosity to understand how the world works. And this has been the case for myself. What I'll be speaking to you about today pertains in part to some research work that my colleagues and myself have undertaken over the last decade or so. So for me, this certainly is a topic that is born from my own sense of curiosity. And of course, I have my brain to thank for that. And then lastly, this topic that we will discuss today is going to be largely one that will relate to primary research discovery. And it's certainly my hope as a neuroscientist that fundamental discoveries will continue to promote healthy living and the treatment of disease. And one of the really fun things about neuroscience discovery is that often basic neuroscience discovery can lead to innovation in the treatment of the human condition in ways that we might not expect as basic neuroscientists. So I think that one is never quite sure where significant application and translation will come from, as long as we continue to pursue our curiosity and to continue to pursue basic discovery in whatever domain of science you may be involved in. And for me, that's especially exciting to be involved in the ever-expanding field of neuroscience. Our learning objectives today are, first, that I want you to be able to discuss the significance of genetic specification, self-organization, and sensory motor experience for the construction of neural circuits in the cerebral cortex. I want you to be able to discuss the significance of experience for the plasticity of neural circuits during what we'll call critical periods of postnatal development. And lastly, I want you to be able to return to a concept that we discussed way back in Unit 2 of the course, and that is Hebb's postulate. And I want you to be able to discuss the relevance of Hebb's postulate for understanding developmental plasticity. Well, I'd like to start our discussion today, as I have the last couple of sessions, and that is by providing a broad overview about the major forces that are shaping brain development. Now, as I think you've been able to appreciate so far, these forces interact with one another, and they do so with one theme perhaps dominating at a different stage of development more so than the others. Obviously, sensory motor experience doesn't become relevant until neural networks are functioning and begin to be modulated by the interaction of the developing child with the outside world. So sensory motor experience is an influence that is emerging in brain development relatively late in the process. Nevertheless, it is an important factor. And what we want to consider today is really, when does sensory motor experience become a major force in brain development, and how does that force interact with these other two, genetic specification and self-organization? Well, just to refresh your understanding of these concepts, genetic specification refers to lineage-derived factors that reflect the spatial and temporal patterns of gene expression. So perhaps it's convenient to think of this as nature, that is, a reflection of the genetic instructions that each cell in the nervous system inherited from its parent cell. Well, self-organization is a relatively underappreciated concept, I would suggest. Self-organization reflects the dynamical behavior of neural systems once synaptic connectivity is established and becomes functional. And self-organization reflects cell-to-cell interactions that are mediated in an activity-dependent fashion. And I'll emphasize activity to describe self-organization rather than experience, because much of the activity in the nervous system is generated endogenously. That is, it is ongoing. It doesn't require external drive to generate that activity. And this endogenous activity provides the substratum, if you will, for self-organization to play itself out. And we'll see a powerful example of that today. Well, sensory motor experience becomes possible relatively later in the story of early brain development as neural systems begin to transduce electrical and chemical and mechanical information that might be present as the developing fetus and developing infant begin to interact with the external environment. And these interactions then produce electrical signals that are driven from external sources and have the capacity then to modulate these ongoing patterns of activity. And all of these factors, genetic specification, experience, self-organization, they interact in complex ways that we still don't quite understand. And the result is the coordinated development of a nervous system that becomes progressively tuned in order to experience the world and respond with appropriate behavior. And this remains much of the challenge of modern neuroscience to understand exactly how this happens. So what I would like to do for much of this tutorial today is to really focus on what we might call nurture. That is, sensory motor experience, the environmental interactions that begin to shape the epigenetics that can modulate genetic specification, and also those modulations that potentially can have a role to play in shaping the outcome of self-organization. So I want us to consider the contribution of sensory motor experience to brain development by focusing in on one key sensory system that has served as one of the principal models for understanding these forces in early brain development. And that model system is the visual system. And I'm going to break this down into really two principal parts. One part will pertain to those properties that are present as the thalamus first supplies inputs to the middle layers of the cerebral cortex. So in this case, we're talking about inputs from the lateral geniculate nucleus that are terminating in cortical layer 4. Now, you'll recall, as we've talked about now since the beginning of Unit 3, that one of the principal architectural features of the cerebral cortex is a columnar circuit that repeats many, many times across a cortical area. And we think that that columnar circuit serves at least three important functions that I've suggested you remember by recalling the ACC of the cortical microcircuit. The first function that's performed by this circuitry is amplification, and that's principally amplification of this thalamic input. So these inputs are processed among small cells that we call stellate cells in cortical layer 4, and from there the information is passed on up into the upper layers of the cortex, and from there down to lower layers of the cortex. So there's sort of a recurrent columnar circuit that amplifies the input that is received in the middle layers of the cortex. We'll go on and talk about the computation of new cortical properties in the next part of this tutorial. So what I'd like to talk about in this first part of the tutorial are the forces that shape the initial topographic mapping of this thalamic input into cortical layer 4. And just to give you a sense of what the outcome of a long series of experiments suggest is that there's an early phase to the development of this system that some have called a pre-critical phase, to emphasize the idea that this phase establishes the basic mapping function that allows the thalamus to map into layer 4 of the cortex, and it's pre-critical in the sense that it doesn't seem to require visual experience in this case, since we will be talking about the visual cortex. Now, this implies then that there must be a later phase that we'll call a critical phase or critical period, where sensory experience, visual experience, can indeed alter the structure and the function of the circuits that are initially established as the thalamus maps into layer 4. Well, as I mentioned, we're going to be talking about the visual cortex. Just a little bit more background to remind you of where we're going with this. So the visual cortex that we have in mind really is the primary visual cortex, which is what we find in the banks of the calcarine sulcus here. This is a beautiful image supplied by my colleagues in our field of visual neuroscience, Dr. Daniel Adams and Jonathan Horton. This part of the brain is exquisitely organized in so many different dimensions, unfortunately beyond the scope of our consideration in this course. But it has captured the imagination and the attention of now at least 3 generations of neuroscientists who've been studying this part of the brain. It is undoubtedly the best understood region of the cerebral cortex, so it serves us well as a paradigm for understanding some of the principal forces that are shaping the development of the brain more generally. Well, this cortex, as you know, is topographically organized. The fovea representation is found at the occipital pole of the hemisphere, and as one progresses from the posterior towards the anterior aspect of this calcarine sulcus cortex, one moves from the central region of the visual field out towards more peripheral regions of the visual field. And this beautiful color code as visual field is related to visual cortex reminds us of this orderly progression of visual topography. It also highlights the fact that what's most important to us in vision is represented by nearly half of the visual cortex, and that is the central 10 degrees or so of the visual world. So it's really the representation of the fovea, and surrounding the fovea, the macula, and then the nearby regions of the retina that account for much of the detail that captures our visual interest. And this is represented by circuitry that spans about half of the visual cortex. That's not to say that what happens in the periphery isn't still relevant for visually guided behavior. Of course it is. But it doesn't require as much circuitry to analyze the details of form and color and location as what we find in the representation of our macular region of the retinas. Now I'll just remind you of what this region of the brain actually looks like histologically speaking. Here's our calcarine sulcus that we see in a histological slide that's stained for the presence of myelin. And on the superior bank of this calcarine sulcus we have the cuneus gyrus, which is going to be representing the contralateral inferior quadrant of visual space, whereas on the inferior bank we have the lingual gyrus, which is representing the contralateral superior quadrant of visual space. Now, this part of the brain is often called the striate cortex because of this densely myelinated stripe of cortex that we see that runs right through the middle layers. So while this stripe is not precisely layer 4, it's found within layer 4 of the visual cortex. So the first part of the story that I'd like to tell you today about the formation of topographic maps and how sensory experience can alter the structure and function of those maps really pertains to establishing the initial connection from the thalamus to this region that's recognizable by this heavily myelinated stripe in the visual cortex. Now you may recall that one interesting feature of this middle layer of the visual cortex that was discovered back in the mid-60s to late 70s is the system of ocular dominance columns that we find in the middle layer of the visual cortex. So let me remind you about ocular dominance. As you know, the two eyes are sending signals into the same lateral geniculate nucleus, but as you'll recall, those signals are not yet combined at the level of the thalamus. Rather, the two eyes project to separate layers of cells within the lateral geniculate nucleus. The contralateral eye projects to layers 1, 4, and 6, whereas the ipsilateral eye projects to layers 2, 3, and 5. It's not yet until these circuits are amplified at the level of the visual cortex do the inputs that are driven from the two eyes first become combined. And in fact, even in cortical layer 4, there's still segregation of these inputs. So as the thalamus projects into cortical layer 4, there is initially a termination of afferents that segregate very quickly into complementary zones, such that one can progress across the visual cortex within layer 4 and encounter these alternating bands or alternating columns, if we think in the radial dimension, that are driven primarily, if not exclusively, just by one eye. Now, once we get above cortical layer 4, the inputs do become mixed. And so this means that stereopsis is first possible in the upper layers of the visual cortex, where information from the two eyes begins to converge on the same cells for the first time in the visual pathway. Nevertheless, even in the upper layers of the visual cortex, there's still a bias towards information that's being driven by one eye or the other. Well, this fact was not lost on David Hubel and Torsten Wiesel as they discovered ocular dominance columns together with their collaborators such as Simon LeVay and Carla Schatz. And what these individuals recognized is that this system of ocular dominance columns in the visual cortex presents a wonderful opportunity to begin to design experiments to look at the role of experience in early life. Well, what was especially powerful about this system is that not only could this system be probed physiologically with microelectrode recordings, and one could assess the degree to which one eye or the other was responsible for driving the action potentials recorded from a neuron in any one of these ocular dominance columns, but there's also an anatomical correlate that could be visualized through various means. And I'd like to show you one other image from my friends, Dr. Horton and Dr. Adams. And this is now one of my very favorite images in the entire field of neuroscience. What we're looking at here is a photo montage constructed from the human visual cortex. And this is tissue that has been prepared and stained to demonstrate the presence of a oxidative enzymes that's found in mitochondria, cytochrome oxidase. And so the intense dark brown regions are those regions that have high levels of cytochrome oxidase activity, and the more pale regions are those with low levels of cytochrome oxidase activity. So the intensity of the brown tells you something about how metabolically active the individual locations in this cortex were in life. Now, this visual cortex was derived from an individual who passed away after having one eye removed. And as a consequence, the ocular dominance columns or stripes in the visual cortex that were driven by the eye that was removed had down-regulated their oxidative metabolism. And as a consequence, the columns that were driven by that eye have now become much less metabolically active. And hence we can reveal for the very first time the complete pattern of ocular dominance columns in the human visual cortex. Now, take my word for it, this was a Herculean effort. Very few histologists are skilled enough to have produced an image such as this. What Dr. Adams and Dr. Horton and their staff were able to accomplish here is truly a remarkable bit of anatomy and histology. What they were able to do is to gently unfold the cortex of the occipital lobe and the upper and lower banks of the calcarine sulcus, to flatten out that cortex and then to produce sections through it that then could be stained to demonstrate this metabolic activity. And what we see is truly a remarkable picture of the structure of the human visual cortex. I hope you're struck, as I am, by really the beauty of this image. So what we're looking at is a representation of visual cortex with the posterior pole of the hemisphere here to the left side of the image, the anterior side of the calcarine sulcus cortex to the right, with the superior aspect, the cuneus gyrus above, and the inferior aspect, the lingual gyrus below. But really the main feature for you to recognize is this beautiful pattern of stripes and columns that we see. So these are indeed our ocular dominance columns, or ocular dominance stripes, as we might recognize them here in this broad view of the visual cortex. So these dark regions are those that are driven by the eye that remains, and the intervening pale zones are those that were driven by the eye that is now removed, and hence they have down-regulated their enzymatic activity. Well, let's consider the physiology now of this part of the brain relative to the inputs derived from the two eyes, and now we'll move to an animal model system where microelectrodes can be inserted into the brain and the two eyes can be stimulated separately, and one can assess to what degree are action potentials driven by one eye's inputs or the other. So what we're looking at here is a histogram that represents the compilation of physiological responses recorded from the kitten visual cortex. The kitten was a very common model system for studying the development of vision back in the 60s, 70s, and 80s. Now we use a variety of other models, including other carnivores such as ferrets, and even rodents such as mice and rats are now common model systems for doing this kind of work. And what can be shown at the level of an individual cortical neuron is that that neuron, regardless of what layer it may reside in the visual cortex, is very likely to have a bias in the response of that neuron that will tell you something about the weighting of inputs that are driven by the two eyes. For example, if a neuron in the visual cortex is driven exclusively by the contralateral eye, then the experimenter will assign it an ocular dominance group of 1. If the neuron is driven exclusively by the ipsilateral eye, then the ocular dominance group assignment would be group 7. So this is purely an operational definition and somewhat subjective, although it can be quantified in different ways by different investigators. Well, somewhere in the middle then would be units that are binocular, that is, they respond to inputs being presented to each eye separately, but they tend to have some bias. So here in the center of this distribution, ocular dominance group 4 would be a totally well-balanced set of inputs being driven by the two eyes converging upon the same neuron. So while that does typically yield the greatest number of cells among these 7 different ocular dominance groups, obviously there are more neurons that are either biased towards one eye or the other than are perfectly binocular. And so this is fairly typical of your normally developing visual cortex, and so it's not perfectly a normal distribution, but it is centered on binocularity with representation on either side. Now in kittens there does tend to be a bit of a leftward shift in this distribution with probably a greater percentage of contralaterally biased neurons than ipsilateral biased neurons. So this is what a normal distribution would look like in a typically developing kitten. Now, the experiment that was initially done by David Hubel and Torsten Wiesel and their colleagues involved keeping one eyelid of a kitten closed during this time of typical brain development in early postnatal life. And what they found as a result of this early manipulation of visual experience is a dramatic shift in ocular dominance bias in the visual cortex. So when they repeated this experiment after surgically keeping shut the eyelid of a developing kitten, what they found was that virtually all of the cells that showed visual responses now became responsive to the eye that remained open. Now, obviously, to do the experiment, they had to open the eyelid that had been sutured shut so they can test whether that closed eye could now drive visual responses, and the answer is that it could not. Now, what was fascinating about this story is that when they recorded from the thalamus of these kittens, they found that the thalamus was responsive to the previously closed eye, but not the visual cortex. So this indicated that there was some change at the level of the cerebral cortex that was primarily responsible for what appeared to be an acquired blindness of that closed eye. And we call this phenomenon of cortical blindness to the inputs presented to one eye amblyopia. Now, yet another remarkable aspect of this discovery was that they attempted to repeat the very same experiment in adult animals, and what they found in adult animals was hardly any change in the relative distribution of ocular bias and binocularity. And when they repeated the very same experiment initiated not at birth but at 1 year of age, what they found was essentially a normal distribution centered on binocularity. So this led to several really important insights. One is that it is possible to change the structure of the circuits that are laid down there in cortical layer 4 as the thalamus innervates the cortex based on the balance of activity that are derived from the two eyes, and that the sensitivity of this circuit to this kind of manipulation of experience is restricted to an early period of postnatal life. And this allowed for the definition of what we now commonly call a critical period. So there is a critical period in early life where these circuits are sensitive to changes in activity-dependent modulation of ongoing neural activity by sensory experience. Now, subsequent investigators went on to ask what actually is different about the circuitry. What was discovered is what appears to be a regression of the terminal arborizations of the afferents from the lateral geniculate nucleus to the ocular dominance columns that are representing the deprived eye. And commensurately, there is an exuberant growth of the terminal arborizations of those geniculocortical projections that are being driven by the open eye. So rather than having a balance of ocular dominance columns, the greater expansion of the cortical territory of the afferents that are driven by the open eye accounts for an increase in the size of the ocular dominance columns that serve the open eye at the expense of those columns that serve the closed eye, which tend to be reduced both quantitatively in terms of numbers of synaptic connections, but also in terms of the spatial extent of their terminal arbors. One might have imagined that following the precritical period, there's some kind of complementary and relatively spatially equivalent distribution of afferents to the visual cortex that are representing the two eyes. And as the critical period begins to ensue and this circuit becomes especially sensitive to the impact of experience, what one might discover is an expansion of afferents that serve the open eye and a regression of those afferents that serve the closed eye. And this is indeed the anatomical picture that was observed by Hubel and Wiesel and Simon Levay, Carla Schatz, and many others since their time doing these kinds of experiments in kitten and, in some instances, rhesus macaque visual cortex. Now, you may be wondering about whether these effects can be reversed, and just about all permutation of this sort of experiment that you might imagine has been done, and I won't take the time to talk about the entire canon of what is known about ocular dominance plasticity in early life, but I will say that it is possible to reverse this effect if we are able to intervene within the critical period. If not, if the critical period passes and intervention does not begin during that critical period while that window is open, this can result in a lifelong visual deficit. This cortical blindness for the deprived eye may persist across the lifespan. So this raises considerable urgency when treating infants that may have a correctable ocular defect in one eye to prevent this kind of amblyopia for setting in and leading to lifelong visual impairment. Well, for those of you that are interested in vision as I am, I hope you find this a fascinating paradigm to learn about. For those that aren't particularly interested in vision, I want you to know that ocular dominance plasticity has remained a gold standard in the field of developmental neurobiology for understanding the influence of experience on brain development in early life, and continues now some 5 decades after the seminal discoveries on this topic were first made. This remains the gold standard in the field of developmental neurobiology. But with any kind of gold standard, one does have questions. One might wonder whether this general lesson that we've learned about studying ocular dominance column development truly applies to all kinds of circuits in the developing brain. Or are there some somewhat idiosyncratic features of this story that might limit our ability to generalize? So what I'd like to do is to suggest that indeed perhaps the latter is the case. That is, we need to explore other kinds of properties of developing cortical networks if we want to understand more broadly the impact of experience in early life, and that what we've learned from studying ocular dominance columns, as powerful as it has been, may not inform us about the development of other kinds of circuits. Well, what I'd like to do is to now turn our attention to the development of other kinds of properties in the visual cortex that probably relate to the emergence and the construction of circuits that are intrinsic to the cortex itself, not just the topographic mapping of thalamus into the middle layers of the visual cortex. So I'll remind you that one important function of this cortical circuitry is to compute new functional properties. So not just to amplify the pre-existing properties that are inherited from the thalamus, but to actually construct circuitry in the cortex that can perform this important function of computation. Now, we talked about a couple of examples of computational properties that seem to arise as much from the operations of intrinsic cortical circuits as does the convergence of inputs from the thalamus. And two of those properties that I'd like to talk about now in the second part of this tutorial are orientation preference and direction preference. So I'll just remind you of what these properties are. Again, the discovery of these properties goes back to the seminal work of David Hubel and Torsten Wiesel. They were rightly awarded the Nobel Prize in 1981 in part for these discoveries. And what they did for the first time was to insert microelectrodes into the visual portions of the brain and record the action potentials from individual neurons. And they were the first to explore systematically the receptive field properties of the neurons that they encountered. And what they discovered when making a penetration in the vertical direction, such as this, is that all the neurons across the depth of the cortex that they could record from all shared similar receptive field properties. That is, they tended to represent overlapping regions of the visual world, and when they tested different orientations or different angles of bar stimuli, what they found is that these neurons all shared the same orientation preference. For example, in this illustration, they encountered a column of cells that all seemed to respond best to vertically oriented stimuli. And as they rotated their bar of light away from vertical, the responses got weaker and weaker, even though they were centered in the middle of the visual receptive field. So this defines what we consider to be the property called orientation selectivity. Now, not shown on this slide is a related property called direction selectivity, and that is that many of these neurons are going to respond to a bar of light that are moving just in one direction, but not in the opposite direction. Or they may at least have a strong preference for motion in one direction or the other. That we call direction preference. So most neurons in the primary visual cortex exhibit these two properties together of orientation preference and direction preference. More recently, newer methods have been developed to complement what can still be done very well with a microelectrode. These are results that I showed you previously in the course using a method called optical imaging of intrinsic signals. And this is a method that requires exposing the visual cortex to light and measuring the amount of light that's absorbed and the amount of light that's reflected. And what we know is that as regions of the brain become active, they increase their neural activity, which consumes more oxygen and induces a hemodynamic response, increasing local blood flow to those regions of the brain that have increased activity. Well, with this method, it's possible then to map out the local regions of the brain down to a few hundred microns that become active. And what's being represented here is the response to a bit of the visual cortex to a set of visual stimuli that vary in the orientation of the elements of that visual stimulus. And what we see when we drive the visual cortex with an oriented stimulus is a series of domains or columns or stripes that become active. And the columns that are driven by, let's say, a horizontal stimulus are precisely complementary to those columns that might be driven, say, by a vertical stimulus. And this complementary spatial location in the visual cortex can be represented by simple image processing methods where, let's say, a horizontal image is subtracted from a vertical image. And when you do that, you get something that looks like this, where now the dark regions are those that are driven by the vertical stimulus, and the white regions are those that are driven preferentially by the horizontal stimulus. And as I think you can tell, these regions perfectly interdigitate. Well, this experiment can be done over and over again for all kinds of combinations of visual stimuli, and we can represent this in a highly dynamic way. So what we're looking at here is an animation constructed by these images that represent the more high contrast black and white view of these interdigitating columns. And what I've done here is I'm showing you representations of the visual cortex from 3 different model species that we've used in the lab. And what I hope that you appreciate from this is really a couple of things. One is the smooth, continuous way that these columns that are driven specifically by given orientations are laid out across the brain. In fact, I think one can even identify that there seem to be regions where these columns seem to circle around one another in what almost look like to be circular points around which there is rotation. Well, of course, there's nothing really moving in the brain. Rather, the dynamic representation of activity is what we see in this animation here. And what this demonstrates is that there are adjacent regions of the brain that are representing slightly displaced orientations of visual stimuli. Well, at least for me anyway, this display can be a bit mesmerizing, especially if one reflects on the architecture of the circuitry of the visual cortex that accounts for these columnar patterns of activity and how they relate to one another as a visual stimulus is changed in a smooth and continuous fashion. Well, let's return to the more static display so we can begin to use this columnar system in the brain as a model system for probing the influence of experience in early life. So I'd like to just mention a couple of conventions because we'll want to use these in the experiments that are designed to assess the role of experience in shaping the circuits that give rise to these columnar patterns. You'll notice that the column that I have here to the left is labeled response. And so what we're looking at here is simply the activity in the brain that represents the presence of the visual stimulus. And to the right, where we have these beautiful dark and light interdigitating domains, we call this selectivity. This is now the result of subtracting one image from another. So the degree to which we see dark and light regions in this difference image, as we call it, is an index then of the selectivity of the cortical response. That is, if the cortex responded uniformly across all columns to the same visual angles, then when we subtract the two, we would expect to see just uniform gray. But the fact that we see these nice high contrast images of dark and light bands and stripes interdigitating with one another indicates that there is a high degree of selectivity where interdigitating columns respond quite strongly to their preferred orientation, and the fall off in activity is rather steep as we move away from their preferred orientation. Well, these data can be combined in different ways, and one way that we like to combine them is via an image processing routine called vector summation that allows us to assign a color to every pixel in the visual cortex. And the color of that pixel is now indicative of what is the best orientation that drives that location in the visual space map. And so what we find in the color code down below is a representation of orientation preference. and the intensity of the color tells us something about selectivity. So for example, where we see these nice intensely red regions, these are areas that responded best and quite selectively to horizontal stimuli. And where we see these intensely blue regions, these are columns or bands of cortex that responded best to near vertical orientations. Well, let's dive into this visual cortex and expand this representation, because I want to highlight the centers around which these columnar structures seem to organize. Now, I would draw your attention to these dark spots, which simply represent the limits of resolution of this method. But around these dark spots are arrayed all of the colors that define this color code for orientation preference. And what the distribution of these colors indicate is that there is a singularity around which circuits that represent all possible orientations are organized. And so we see this marvelous degree of organization repeated many, many times across the visual cortex. We call these point singularities pinwheel centers, and the complete structure that contains the columns that represent all possible orientations for a given set of overlapping receptive fields, we call those pinwheels. So the visual cortex then is organized with the circuits that take the shape of pinwheels, and these pinwheels repeat many, many times across the surface of the cortex. And in some work that I was privileged to participate in with some collaborators who are theoreticians and computational neuroscientists, what we were able to do is to examine a large set of orientation preference maps from different species. And we did a fairly simple analytical procedure, although it was not trivial to perform this. What we did was we simply assessed the density of pinwheels in the visual cortex. And when we plotted the density of pinwheels in units of area of measurement and related that to the overall size of the pinwheel, something that we call the hypercolumn size, what we found is that all the data points seem to follow along a single line. And when we simply divided these two axes from one another, that allowed us to generate a dimensionless expression called pinwheel density. What we found was that now all of these data points fall along a horizontal line. Well, what was especially exciting about that is that it suggests that there may be some kind of universal principle that describes the way this circuitry organizes in development. And working with some computational scientists, it was easy to get excited about exactly what this value was representing. What we did was we measured a very large sample and discovered that the average pinwheel density across our sample was, well, 3.14, which is statistically identical to the mathematical constant pi. In fact, our confidence interval, shown in parentheses here, indicates that pi is within our confidence limit of our ability to make this measurement in real brains using relatively imperfect experimental methods. So what should it mean that circuits in the visual cortex organized to form pinwheels, and the density of those pinwheels evidently is pi across a broad range of mammalian species? Well, we think this is a signature of self-organization. And some modeling experiments that ourselves and others have done indicate that there are really two important factors that account for the self-organization of these networks. One important factor is that these networks are dynamical systems, and they're subject to activity-dependent plasticity. Another important factor is that these networks grow out long-ranging axonal connections that are able to bind together the activity of different cortical columns that develop similar preferences. We think that these two factors together help to explain why the visual cortex organizes itself into these pinwheel formations with a density of pi. Well, I tell you all this so that I can describe for you some experiments that my colleagues and I have done to look at how the experience of the animal in early life impacts the self-organization of these cortical networks that perform these computations that account for orientation selectivity and direction selectivity. And we began by simply looking at the emergence of these orientation preference maps in the visual cortex of animals beginning from just before the time that they naturally open their eyes and then on through the phase of normal cortical development. And so if one looks at an animal soon after it has opened its eyes, one can indeed find that there is a system of orientation columns that are organized into a map of orientation preference. So this has been known since really the early 90s, when it was first possible to apply these methods to the developing visual cortex. And what was discovered is that these columns do indeed self-organize. They appear pretty early in postnatal life. In those animals that are born with their eyes closed, they seem to emerge prior to the time of eye opening, but they rapidly develop in the days to weeks that follow the onset of normal visual experience. So we thought an obvious experiment to test whether vision had an important role to play in the maturation of this system was to raise animals in total darkness, and thus removing visual experience from the life history of the animals. And when we did that experiment, what we found was that, sure enough, the circuits for orientation, selectivity, and preference do self-organize. It's possible to recognize columns in the response maps and in the selectivity maps that we generate from these animals. And the signals are sufficiently robust that we can assign color values and create a orientation preference map. And with the proper analysis applied to these maps, indeed, these maps from animals that never saw any photons of light until the experiment do indeed self-organize and develop pinwheels with a density of pi. However, I think perhaps even your eye can pick this out. The contrast of the images that we recorded from these animals that lacked vision seemed to be significantly less than what we observed from animals that had normal vision throughout the period of brain maturation that we studied. So while these cortical networks do indeed self-organize without the benefit of vision, vision does seem to contribute to the refinement of these maps and an increase in the selective responses of the neurons whose signals we're recording with this method. Well, just because these circuits self-organize and are present in animals that have never experienced vision does not mean that this process cannot be influenced by experience. So we thought it was important to do a complementary experiment. Rather than just taking away vision, we thought we should make vision abnormal. And a fairly straightforward way to do that is to raise animals keeping their eyelids shut. And that's very much like what your eye might do if we were to close our eyes and then look around the visual scene. If I look up towards the fluorescent lights in the ceiling, I can tell that I'm looking up and seeing an increase in illumination compared to if I were to look away. But I see no form, I see no structure. I see no shape to what I view, simply an increase or a decrement in light intensity. So this is the nature of visual experience that we imposed on a set of animals. And when we did this, what we discovered is that there were some pretty significant impacts on the development of the circuits that compute orientation preference. Now, this manipulation did not produce cortical blindness. And we know that because when we looked at the response maps in the visual cortex, we saw robust activation of the visual cortex. The problem, however, is that we fail to see the robust development of columnar structure. It's as if the very same circuits in the brain responded to the presentation of a horizontal stimulus and the presentation of a vertical stimulus. So when these images were subtracted, what we found was virtually no evidence of columnar structure that was organized into a map of orientation preference. So I want you to notice the implication here of this finding here. What this shows us is that when we made vision abnormal, what we found was actually more significant impairment of the visual cortex than when we simply deprived the animal of vision. Now this reminds me, at least, of a famous aphorism that was suggested by one of our seminal neurosurgeons of the 20th century, Dr. Wilder Penfield, who said that no brain is better than bad brain. Well, that justified some neurosurgical procedures aimed at the removal of bad brain. In this case, I would suggest that bad experience is worse, more detrimental to the development of visual cortical circuits than is no experience. So what have we learned from these studies? I think we can make some provisional conclusions based on these experiments looking at the development of orientation preference. I think what we've learned is that normally circuits in the visual cortex self-organize, and they operate synergistically with normal sensory motor experience. And this synergy promotes the full maturation of these circuits. However, when experience is rendered to be abnormal, then this synergy is broken and self-organization goes awry. The neural circuits that develop as a consequence are functionally impaired. Now these neural circuits, they self-organize to adapt to the quality of the incoming sensory signals. So by self-organization going awry, I mean relative to the synergy that our brain has evolved to anticipate from the world in which we live. But when that synergy is abrogated by some kind of ocular impediment, in our case keeping the eyelids closed, or perhaps in clinical populations, congenital cataract, for example, would be one human condition that we would model through this method of keeping the eyelids shut. Well, such conditions, keeping the eyelids shut or congenital cataract, are those that would provide for the adaptive influence over the circuitry of the visual cortex that would then develop. So consequently, not only do these neural circuits under these conditions fail to benefit from normal visual experience, and as a result, they're actually developing along a trajectory that instantiates functional impairment. So now I'd like to turn our attention to the property of direction selectivity and direction preference. So we can think of this as a further differentiation of the circuitry for orientation selectivity and orientation preference, where those circuits begin to specialize for the representation of motion in just one direction or another, with the preferred direction of motion being orthogonal to the preferred orientation. That is to say, circuits might develop a preference, let's say, for vertical stimulus, but there might be a differentiation of one side of the orientation domain to prefer vertical moving to the right, whereas the other side of that orientation column might differentiate a preference for vertical moving to the left. Now we can resolve this with the same method I've just described for you, this optical imaging of intrinsic signal methods. One can even resolve it to the cellular level using a method known as two-photon imaging of calcium signals, which is what is represented here to the right. So it's now possible to do studies where, for example, we can zoom in on a small region of the visual cortex and recognize that with this more low-resolution method, we can define a region that responds, let's say, mainly to downward motion. And that region representing downward motion seems to be separated by some kind of barrier condition from an adjacent region that represents upward motion. We imagine that within this hypercolumn here, there is a orientation column that represents horizontal, but it has subdivided into these two adjacent domains that represent opposite directions of motion. And then zooming into this region with this method that provides cellular resolution, we see indeed there is a population of cells, each cell being represented by an arrow here, that has a preference. And the upper right region of this field seems to show a predominant preference for upward motion, whereas the lower left region of the field shows a predominance of cells that prefer downward motion. Now, my colleagues and I were quite interested to understand how do these two properties of orientation preference and direction preference develop conjointly in the very same animals. And we thought that the way to begin to study this question was simply to follow the development of a cohort of animals that were raised under our normal laboratory conditions. And when we did that, what we discovered is displayed here in this slide. What I'm showing you are representations of orientation selectivity and direction selectivity acquired from the very same animal within this cohort. And what we did was we sampled these animals at different stages of development following the time of natural eye opening, which is right around the end of the first month of postnatal life. What we found is that when we looked at animals right around the time of natural eye opening, orientation preference was present, as indicated by this series of orientation columns interdigitating with one another. But there was virtually no evidence of a differential response to opposite directions of motion. That began to be visible after a few days of visual experience following eye opening, but it really wasn't until about 7 to 10 days later that we began to see this differentiation of our orientation columns into domains that prefer opposite directions of motion. So the upshot here when we looked at the developmental curves is that while orientation preference is present at or just before eye opening and continues to develop throughout the first few weeks of life, there seems to be a real lag in the development of direction preference. There seems to be about a week or so of visual experience that's necessary before there is a steep inflection in the development curve. And then after about 2 to 3 weeks of postnatal life, at least in this animal model, mature levels of orientation and direction selectivity are established. So the fact that there seems to be this lag in the development of direction preference suggests that perhaps vision in this early period of postnatal life is actually doing more for the development of direction preference than it is for orientation preference. Well, in order to test this idea, we did similar kinds of experiments, raising animals under normal conditions and comparing them to animals that were raised without vision. And here are some data that represent the cohort of animals that were raised with normal visual experience. And these animals develop robust cortical maps for orientation selectivity and direction selectivity. And these symbols here represent the measurements that we were able to obtain from these animals, reflecting the degree of development of selective responses to visual stimuli that varied in orientation and direction of motion. So this is a picture of what normal development looks like. When we raised animals in darkness, as I've just told you, these animals do in fact self-organize a map of orientation preference, but the degree of selectivity is less than what we would see with the full benefit of visual experience. Well, what was really surprising to us, and actually quite unexpected, was that there was virtually no sign of the emergence of direction selectivity in these animals. In fact, quantitatively speaking, these symbols are clustering around 0, which is equivalent to the absence of a visual stimulus. And this was quite a remarkable dissociation of the development of the circuits that compute direction selectivity from those that are responsible for at least the essential computations that compute orientation selectivity. Well, we wanted to know at what time in development was this network especially sensitive to the onset of visual experience. And what we found is that if we deprived animals of vision for the first couple of weeks after eye opening and then gave them what we might call late vision, we found that their orientation preference maps returned to their full developmental strength. And you can see that by eye with the robust dark and light distribution of orientation domains in the maps from these animals. However, the data that represented the development of direction selectivity really remained no different than those that were completely deprived throughout the developmental epoch. In fact, these data sets are no different from one another, and statistically speaking, not different from the comparison of maps taken from animals with no visual stimulus. So what we found is that there remained severe impairment in the circuits that would compute direction of motion in the absence of early vision. In fact, it's really only those animals that receive the benefit of early vision that then go on to develop direction domains indicative of maturation of circuits that are computing direction selectivity. This cohort of animals were deprived of vision up until the time of natural eye opening and thereafter given a couple of weeks of visual experience. And these animals are acquiring a strength or robustness of visual computation equivalent to animals that were simply raised under normal laboratory conditions right from birth. So these data suggest quite powerfully, I think, that the circuits that compute direction selectivity are indeed more sensitive to early vision than are those circuits that self-organize in order to compute orientation selectivity. And it suggests that there is a very early critical period in visual cortical development when normal visual experience is essential in order to develop the circuitry that allows us to compute movement to one direction or another. You can imagine how important that is for survival in a world that is full of objects that are moving. And indeed, as we move through the world, we generate our own patterns of visual motion in the visual cortex. Because there is this very early critical period then for the development of these circuits, we wondered if it might be possible to actually train this circuitry with structured vision. And so this allows us to get at this same problem really from a complementary direction. Rather than depriving vision, we wanted to know whether we could provide some structured vision and impact the developmental course of the circuits that are necessary to compute this property. And indeed, that turned out to be the case. So what we did is we prepared some animals for our experiments, and while they were anesthetized and prepared under stable physiological conditions, we were able to expose them to a regiment of visual training. And we asked whether it might be possible to actually observe the development of these circuits in the brain. And indeed, as we looked over an experiment that lasted about a day, what we saw is that after about 8 to 10 hours of visual training, we began to see the emergence of structure in our selectivity maps for direction. Indeed, we begin to see the emergence of these light and dark domains that are beginning to take shape in a fairly robust way after about 12 hours of visual training. So what was especially remarkable to us is that at the end of the experiment, when we tested these animals for their responses to all possible directions of motion, what we found is that the differentiation of direction domains was really only present along the axis of motion that was used for training purposes. So this provides perhaps some of the best evidence that we know of in the field of neuroscience that visual experience can actually instruct the development of circuits in the cerebral cortex. Now we've gone on to study this phenomenon at the cellular level using this method I mentioned a few moments ago, two-photon imaging of calcium signals. And this has allowed us to watch cell by cell the emergence of direction selectivity and direction preference in the visual cortex. And what we're able to do is to follow the same cells over time. Here we're looking down in a very small region of the visual cortex. And after about 3 hours of visual training, we can observe the very same cells in the living animal. Here we can identify many of the same cells that are giving rise to a fluorescent signal after we've provided a dye that is taken up by these cells and can report neural signals based on calcium fluxes that are present within these cells. And in the initial characterization, what we find are fields of cells that have some degree of direction preference. So these are 3 different animals here, and what we're looking at is their responses and preferences before training and then after about 3 to 6 hours of training. And what I hope you can appreciate is that in each of these cases, there is an increase in the numbers of neurons that prefer the direction of visual training. In this top case, training was done with motion to the left, and what we see is an increase in the numbers of neurons that prefer that leftward motion. In the middle case, the training stimulus was moving down to the right, and we see a modest increase in the numbers of cells and the strength of selectivity indicated by the size of the arrows in this field of neurons following this bit of training, with a bias towards movement down and to the right. And the same is true in the third case, which already had a fairly significant bias of cells that preferred rightward motion. And then after 3 to 6 hours of visual training, those cells became even stronger in their preference, and a few more emerged. So when we looked at our entire data set before and after, we saw indeed a statistically significant increase or a rightward shift in this cumulative plot of neurons that acquired the direction that was presented by the training stimulus. So what has this taught us about the role of visual experience in the development of direction preference? Well, what we've learned is that the neural circuits that underlie direction columns don't seem to have the capacity to self-organize in the way that those circuits that were responsible for orientation columns self-organized. Rather, the differentiation of those circuits to compute direction selectivity must be instructed by the presence of a visual stimulus. We've also learned that the window of opportunity for the benefit of this motion training is very brief. So this means that early experience is absolutely critical to avoid what might be a long-lasting impairment in the ability to differentiate directions of visual motion. I would suggest that in order to understand the construction of neural circuits in the brain, we need to examine multiple circuits that exist at different stages of cortical processing. We shouldn't just rely on one gold standard, despite how valuable that standard might be for a certain line of investigation. We should always be cautious about generalizing the principles that we've learned from just one model system to another. We should be somewhat leery of this two-phase view of development, that is, this idea of a pre-critical period followed by a critical period. I think that for many circuits in the brain, I would suggest that this two-phase view will not be adequate to account for the influence of experience on brain development. Thus, sensory motor experience can influence the construction of neural circuits as soon as the brain becomes responsive to environmental signals. So to put this all together, I would suggest that genetic specification, self-organization, and sensory motor experience interact concurrently, not just sequentially, but as soon as all of these influences could possibly interact with one another, they are competent to do so. And this interaction shapes the ongoing development and refinement of neural circuits in an early phase of life, and potentially across the entire lifespan. Well, what are some implications of these findings? I would suggest that one implication that should not be lost on us is that normal sensory motor experience has a profound effect on the formation and the maturation of neural circuits in the cerebral cortex, especially those circuits that reflect the computations performed by networks of neurons that sit just beyond the early input layers of the cortex. There very likely are many properties of cortical networks that depend upon sequence or timing-based signals, like direction selectivity, that may not develop at all without the benefit of normal experience in an early critical period. And this is perhaps the strongest implication of the studies that I've showed you today. I think what we've discovered is that abnormal sensory motor experience in these early critical periods can lead to lasting functional impairment, and that abnormal experience might be the absence of experience, or it more likely is going to be experience that is filtered through some kind of peripheral abnormality in the formation of the sensory organ or in a musculoskeletal unit that will impact the way that organ is operated in early life. So to sum this all up, I would say that normal sensory motor experience in early life is truly critical for lifelong health and wellness. In the final segment of this tutorial, I'd like to return to thinking in a more principled way about the relationship between early life experience, patterns of electrical activity in those developing neural circuits, and then the influence of neurotrophins. Well, do you remember Hebb's postulate? We talked about this when we first considered synaptic plasticity back in Unit 2. Hebb's postulate states that the coordinated activity of a presynaptic terminal and a postsynaptic cell would strengthen the synaptic connection between them. And conversely, uncoordinated activity between synaptic partners would weaken their synaptic connections. Well, these aspects of Hebb's postulate, I think, is very aptly summed up by my late great colleague here at Duke, Dr. Larry Katz, who said that neurons that fire together wire together. Now let's try to relate what I've been describing for you as the important role of sensory motor experience in cortical development in terms of Hebb's postulate. Well, Hebb postulated that what was important for the development of strong connections is the coordination of activity. Well, I would suggest that one important source of coordination is normal sensory motor experience. So this kind of experience can structure the activity in sensory and motor pathways and provide an important source of spatial and temporal structure that might essentially provide for the coordination of pre- and postsynaptic elements. So imagine, for example, the developing visual system that's encountering patterns and objects that are defined by the orientation of their boundaries. Some of these objects are moving relative to others. This provides a rich source of spatial and temporal cues that can drive the coordination of firing in the circuits that are being constructed in early life to compute properties such as orientation and direction selectivity. So for those circuits that are being activated, we imagine that there's coordination between the presynaptic and postsynaptic elements. Very likely we're satisfying our spike timing dependent plasticity rules for the strengthening of synaptic connections. That is, for those circuits that are being shaped by this activity, there's presynaptic activity that is driving postsynaptic activity. Those circuits where the presynaptic input fires prior to the postsynaptic cell by a short period of time are likely to be reinforced. Now consider the impact of sensory deprivation, be it total deprivation or perhaps abnormal deprivation. One way to understand the impact of abnormal experience is that we are failing to provide the spatial and temporal cues that are essential for sculpting activity within these developing cortical networks. Perhaps we are failing to drive the successful coordination of pre- and postsynaptic elements. And with essentially the introduction of noise into the activity patterns of these developing networks, I think we are more likely to favor uncoordinated activity between pre- and postsynaptic elements, with more often than not a postsynaptic neuron firing in advance of any given presynaptic input. That might favor the depression of synaptic connections, perhaps even the elimination of synaptic connections that otherwise would have anticipated normal experience and a synergy that would allow for self-organization and the impact of experience to work hand in hand to build up functional neural networks. If anything, the neural networks that are built up will be adapted to the abnormal experience that's driving activity in that network. Well, Hebb's postulate allows us to account for the early impact of shaping activity within developing neural networks, but the long-lasting impact is going to require additional molecular signals, and perhaps that's where neurotrophins come in. So let's try to weave into this story what we learned in our last tutorial about the importance of neurotrophins for promoting the growth and survival of neurons and their connections. One important point to emphasize is that the production and release of neurotrophins is activity dependent. Secondly, that neurons that are connected and coordinated would likely strengthen their interconnections and be nourished via the release of neurotrophin in an activity dependent manner. And that neurotrophin will be taken up by the presynaptic elements and will have local effects on those terminals that are binding neurotrophins to their receptors. But the internalization and the retrograde transport of those endosomes back to the cell bodies can have a long-lasting impact on the viability of the cell that is being so nourished. So in this way, we think that neurotrophins become an important mediator of the long-lasting benefits of coordinated activity between pre- and postsynaptic elements in developing circuits. Now, the converse might also help us to understand the impact of abnormal experience. So for those circuits that are driven by abnormal experience, that is, there's functional connectivity but poor coordination, these connections will be weakened as a consequence of the conditions that would favor long-term depression. But these connections are also going to be weakened and eventually eliminated because they will fail to acquire this neurotrophic substance that is released in limited quantity. So these connections are going to fail to thrive because they're being deprived of sufficient neurotrophic support. Now we can extend these ideas really across the lifespan and suggest that even in maturity, neurotrophins become an important mediator of plasticity as the brain continues to adapt to sensory motor experience. So in the mature system, the regulated secretion of trophic factors continues to help shape the structure and function of neural connections, and this is especially true in response to injury or to adaptations to new patterns of experience, the kinds of patterns that might be engendered as we try to learn a new skill, or perhaps as we try to accept the challenge of learning medical neuroscience. So hopefully hanging with me through these many hours of tutorials and all the great studying you've done on your own has promoted the release of neurotrophins in circuits that are helping to make you a more efficient and effective learner, and hopefully to learn in a way that allows you to apply this new knowledge in a variety of settings. Now, in the final tutorial in Unit 5 on the changing brain, we're going to focus a little bit more on how the brain is responding to injury and disease. But one factor that I want to help you understand is that In the brain that's dealing with some kind of injury, neurotrophins have an important role to play in promoting adaptive plasticity. So in the recovering brain, we know that the activity of certain neurotrophins like brain-derived neurotrophic factor, BDNF, this has been linked to adaptive plasticity in circuits that are mediating functional recovery. What I find especially fascinating are recent genomic studies that suggest that as human beings, there are variations in our BDNF gene that might render cortical circuits more or less adaptive to motor challenges or cognitive challenges, and especially in our capacity to respond to injury. So neurotrophins may provide us with a wonderful therapeutic opportunity to increase the adaptive plasticity that might facilitate functional recovery following injury to the brain. So I'd like to conclude this tutorial with one thought that sort of drives these basic science concepts into clinical practice. And whether you aspire to a career in the health professions or find yourself there already, I want you to know that whatever you do with your patients and clients that are living with brain injury, that is, the kind of sensory motor experience that you might prescribe for your patients in the course of their activities of daily living or a more structured program of rehabilitation. This is powerful. This has the potential to modulate the production of neurotrophins and shape the plasticity of circuits, be it a child with ongoing development and construction of neural circuits or an adult where developmental programs are being re-engaged and expressed anew as a compensation of the brain to injury and disease. Well, I do a fair amount of my teaching here at Duke with students in the allied health professions, in particular with students that are becoming doctors of physical therapy. And I love to talk about this point with that group of learners in particular, because for those groups of students, they're entering a profession that neither medicates the nervous system nor does it cut the nervous system. And I might suggest that they have opportunity to do something even more powerful. They get to structure the sensory and motor experiences of the human body as a therapeutic modality that has the power to alter the structure and function of circuits in the brain and the spinal cord. Even without pharmacological intervention or surgical intervention, it's possible to shape ongoing patterns of activity that can promote functional adaptation. And the more we learn about these basic neuroscience principles of plasticity and the interactions among these various forces that shape the structure and function of circuits in the brain, the more effective can our interventions be. Well, thank you for hanging with me in this tutorial and for giving me an opportunity to share in some measure of detail some of the research findings that I've been privileged to be part of over the last decade or so. I think the upshot for me, and I think for most of my colleagues in the world of developmental neurobiology, is that we're entering a wonderful era where we are discovering more about the rules that govern the plasticity of circuits in early life, and it's empowering us to understand best how to promote the adaptation of circuitry to maximize health and wellness, especially in critical periods of brain development. And we trust that what we are learning about early brain development can apply throughout the lifespan, especially in the context of neural circuits that are adapting to injury or disease. So one of the great promises of developmental neuroscience is the discovery of basic principles that can apply throughout life as the brain re-engages programs of brain development as a means of coping with disease and injury. In our final tutorial in Unit 5 on the changing brain, we will focus on how the brain continues to change out of the early years of postnatal life through adolescence and into adulthood. And we'll spend much of our time thinking together about how the nervous system responds to injury and disease. So I look forward to that final discussion with you that will put a conclusion on Unit 5, and that will allow us to progress on into the final stage of the course as we embrace the complex brain functions that will be the focus of Unit 6. So I'll see you there shortly. Welcome to the final tutorial in Unit 5, which is all about the changing brain. And today's tutorial is entitled The Changing Brain Across the Lifespan: Development, Repair, and Regeneration. Today's tutorial relates to two of our core concepts in the field of neuroscience. First, that life experiences change the nervous system. We'll spend some time in this tutorial talking about a variety of life experiences that have an impact on nervous system structure and function, including injuries to the nervous system. And hopefully what you'll see is that fundamental discoveries promote healthy living and the treatment of disease. And the more we know about the basic mechanisms of nervous system change and plasticity and the response of nervous tissue to injury and disease, the more we are likely to develop strategic interventions that will do just this, promote healthy living and the treatment of human disease. Well, I have several learning objectives for us in this tutorial. First, I'd like for you to be able to discuss the neurobiological basis for changes in gray matter and white matter volume that occur in the developing brain throughout the years of childhood. I'd like for you to be able to discuss the mechanisms of regeneration of peripheral nerves following injury. And I'd like for you to be able to discuss the mechanisms of plasticity in adult sensory motor maps following peripheral injury. I'd like for you to be able to discuss the mechanisms of plasticity in adult neural circuits following central injury, that is, injury to the brain or the spinal cord itself. And lastly, I would like for you to be able to have a cogent discussion of the evidence regarding neurogenesis in the adult central nervous system, and that will include evidence for neurogenesis as well as evidence against neurogenesis. Well, we will begin this tutorial as we have the last several, just recognizing that there are three principal forces at work to shape brain development. Certainly these are powerful forces in embryogenesis and in early life, but they continue to interact with one another throughout the lifespan as genes are expressing their protein products. So there's the contribution of nature or genetic specification. Cells continue to interact with one another in a dynamical way through activity-based mechanisms, whether that activity is wholly endogenous or modulated by experience. So there's the potential for self-organization to continue to play out. And of course, we interact with the environment through our sensory and motor systems, and this allows for these endogenous patterns of activity to be shaped by the stimuli that we encounter, the way that we actually use our body. So this would be sensory motor experience. And what is particularly interesting to consider, and I wish we could do more, but our knowledge is limited, and so is our time, as you all well know. These three factors, I think, become especially intriguing and important to consider when we think about how the brain responds to injury. For example, we might imagine that when the brain is injured, then the dynamical system of that network is perturbed in some way. And this very likely is going to alter the way these ongoing patterns of activity are shaped by interactions with the environment. Likely, these dynamical systems will fluctuate and have an impact on the expression of genes, perhaps the expression of genes that have been dormant since the early days of embryogenesis. And likewise, the gene products then will interact with our neural systems that will affect the way they transduce electrical impulses and encode information about the conditions of the body and the outside world. So these three factors continue to shape how the brain responds to injury. So that's just a thought I'd like for you to keep in your mind as we consider some of those contexts later in this tutorial. But let's begin by picking up the story of the changing brain across the lifespan, where we left off last time, which is in neonatal life, at a time when the brain is especially sensitive to the impact and the influence of nurture or environmental interactions, be they for the better or for the worse. So I'd like to continue the story of development and extend now the context out throughout childhood. And one very important consideration that we should mention as we consider how the brain changes in childhood is the creation of new synaptic connections. And we call this synaptogenesis. And there had been a variety of reports about synaptogenesis in the human brain throughout the 70s and 80s, and I think largely these data were somewhat flawed simply because the methods just are extremely difficult to employ successfully in human tissue. So no fault to the investigators, but just that we have not yet developed the methods that allow for the generation of reliable data on this front. Well, there was a heroic series of studies led by Pachko Rakesh at Yale University and his collaborators and his students over the years that systematically looked at the density of synaptic connections in an animal model, a very excellent model for understanding synaptogenesis in our brain. They looked at the rhesus monkey, and they examined a variety of cortical regions over a series of studies that spanned the decade of the 90s and into the early 2000s. And the results of these studies are summarized in this slide here, which is taken from our textbook. And what is being plotted here is the density of synapses across early life in the rhesus monkey. And what is very clear from these data is that in all the cortical areas that were examined, this includes primary sensory cortex, motor cortex, dorsolateral prefrontal cortex, and cortex that's close to the limbic forebrain of the medial temporal lobe. What's very clear is that at the time of birth, there are relatively few synaptic connections that are established and fully functional. Thereafter, however, there's an explosive increase in the numbers of synaptic connections. So we see that in all of our cortical areas that have been studied. In the months after birth, and this might correspond to the first couple of years of infancy in humans, there's a tremendous increase in the numbers of synapses that are being proliferated in the cerebral cortex. Now, that's not to say that there may not be some loss of some synaptic connections. Indeed, there will be loss of some synaptic connections, but rather it's to emphasize that in early infancy, there is a net gain in synaptic connections by at least 2 or 3-fold over the numbers of synapses that we're born with in the cortical mantle. So it's reasonable to conclude then that part of critical period plasticity, where the brain is especially sensitive to the influence of the environment, has to do with this phenomenal rate of synaptogenesis that we see during the onset and near the peak of the critical periods in each of these areas. Well, for a variety of reasons, we know that the critical period does indeed begin to wane after the first few years of life. Here's just an example of an apparent critical period for the acquisition of the English language. Compared to native speakers, the development of fluency begins to fall rather steeply after the first few years of life, as is shown here in this plot. So after about age 7, there is now a progressive decline in the relatively fluency for those individuals for whom English is not their native language and they are trying to acquire English language skills. Now, before I say more about this decline of the critical period and what happens as synaptogenesis wanes, I should point out that in this early constructive phase, when synapses are being added at a phenomenal rate, is an opportunity for brain development to go in a different direction, a direction that may be related to various kinds of learning disabilities or other kinds of disorders of cognition. Let me give you just a few examples of this. What seems to be known about individuals that are somewhere on the autism spectrum disorder is that there appears to be an acceleration of this capacity for synaptogenesis, such that if one simply looks at overall brain size, in children that are beginning to be evaluated and diagnosed with autism spectrum disorder, it's very possible that there's actually an acceleration of the rate of synaptogenesis in their cerebral cortex, which results in an overgrowth of the brain, especially involving the cortex of the frontal and the temporal lobes, as well as the amygdala, that we'll say more about in the next unit of the course, as well as the cerebellum. And these are brain structures that are involved in social cognition, communication, and the executive control of motor actions. Now, as development proceeds in these children, the overgrowth tends to normalize towards where a typically developing child might be. But that's not to say that connections are restored to normal structure and function. It very well may be that aberrant connections that may have been developed during this period of overgrowth may persist throughout the childhood and adolescent years, and indeed even into adulthood. Now, I'll give you just one other example, and that's the example of attention deficit hyperactivity disorder. In this disorder, commonly known as ADHD, rather than there being an acceleration of synaptogenesis, there seems to be a decrease in the rate of synaptogenesis, such that the cerebral cortex, again, mainly in the frontal lobe and in parts of the temporal lobe, seems to be thinner than typically developing children that don't have this disorder. So it very well may be that this decrease in cortical thickness is attributable to a relative delay, or perhaps a relative reduction in the total numbers of synaptic connections that are established. We still don't really understand the full consequences from a functional perspective, but it's reasonable to at least hypothesize that this reduced rate of synaptogenesis is leading to the construction of dysfunctional circuits that can modulate behavioral control. And perhaps this is part of the phenotype associated with this particular disorder. Now let's just back up to the data that illustrate for us the rates of synaptogenesis as indicated by the synaptic density that's found in the cerebral cortex. And I'll highlight now, after this initial dramatic rise in synaptogenesis, that we in each of these cortical areas see a reduction in synaptic density that follows in the subsequent years, well, for humans, for the first decade of life, let's say the second half of that first decade. And so what we see is anywhere from about a 20 to 50% reduction in the numbers of synapses that are found in the adult cerebral cortex compared to this time of peak synaptogenesis, which is probably sometime near the middle of the first decade of life. We think that one consequence of this reduction in the synaptic density in the cerebral cortex is an overall reduction in the volume of gray matter that we see throughout the latter childhood years and through adolescence on into adulthood. Now, here are data that show us gray matter volume obtained from MRI studies, showing data broken down by males and females. The average male brain is larger than the average female brain, so it's not surprising that the average thickness of gray matter of males should be greater than females. But both males and females show essentially the same trend, and that is a reduction in the volume of gray matter throughout the childhood and adolescent years. Again, presumably reflecting not a loss of neurons necessarily, but a loss of synaptic connections. Now again, one can hypothesize that this process might actually be normal and physiological, but it also may go awry in certain developmental disorders. So there are data on this issue from several developmental disorders and cognitive and behavioral dysfunctions. In attention deficit hyperactivity disorder, for example, it appears that not only is there a reduction in the rate of synaptogenesis, but there may be a modest acceleration in the loss of synaptic connections. such that in children with a diagnosis of ADHD, they may end up with a significantly thinner cerebral cortex, again, especially in these frontal executive networks as well as in the temporal lobe, compared to typically developing children. Now, this difference is not great, but it is detectable in cohort studies, and it suggests that there may be something really quite different about the wiring of neural networks in the cerebral cortex of children with this diagnosis. Now, one interesting aspect of brain development throughout these childhood years is that although the volume of gray matter is getting reduced over time, the volume of white matter is actually increasing. And again, this is the case for both men and for women. And there's a lot of activity trying to understand exactly what accounts for this increase in the thickness of white matter. One can reasonably assume that a principal factor is an increase in the myelination of axons in compact white matter pathways that are found within the brain. But there are other possibilities that likely also to contribute, such as an increase in the diameter of axons, potentially even an increase in the numbers of axons that are present in white matter pathways, reflecting ongoing construction of circuits that might continue to take place throughout the first few decades of life. In addition, there may be an increase in the extracellular spaces in this white matter, or an increase in extracellular matrix that would contribute to this overall increase in volume. So these are all open questions, and I'm sure we will learn much more about this in the next couple of years of study. Well, let's now carry on this discussion beyond the first couple of decades of life and look at what happens to overall brain size across the lifespan. As has been now demonstrated in numerous studies, the human brain increases rapidly in the first few years of life, and brain size seems to reach a bit of a plateau somewhere around the beginning of the second decade of life. And then throughout the second and into the third decade, there's not much change. But beginning in about the third decade and beyond, there is a gradual decline in the overall size of the human brain. So again, gray matter is getting smaller rather rapidly towards the end of that first decade, and then into the second decade, whereas white matter is expanding. But by the time of the third or fourth decade, there seems to be a net reduction in both gray matter and white matter volume that continues across the lifespan. There may be some parts of the brain that are changing more rapidly than others, perhaps some white matter pathways that are changing more rapidly than others. One common idea in cognitive neuroscience is that the white matter of the frontal lobe is the last region of white matter to become fully myelinated or to achieve its full volume. But it may also be the part of the cerebral white matter that is the first to show reductions in white matter volume, perhaps reflecting a loss of myelin or disordering of myelin that continues across the lifespan with increasing rate of disruption of myelin with advanced age. Again, further topics that require much more study for us to fully understand exactly what's going on with the aging brain. Now, one last point that I would make about this decline in brain size is that it seems fairly clear that this reduction in overall brain size is not due to a loss of neurons, but rather through these kinds of reductions in myelin and reductions in the numbers of synapses in gray matter that lead us to hypothesize that there is a reduction in the connectivity of the brain rather than the numbers of neurons per se. If neurons are being lost, then we attribute that to a neurodegenerative pathology. So this is a bit of a changing concept in aging neuroscience, where we now typically understand that the loss of neurons is pathological and should not be considered part of normal aging. Okay, I think we're ready to switch gears here and talking about how the brain changes across the lifespan. I'd like to turn our attention to a couple of specific contexts, beginning first with the plasticity of sensory and motor maps in the brain, where these plastic phenomena have been best studied and best understood. So what we know is that the somatic sensory and somatic motor maps can undergo a fairly dramatic reorganization across the lifespan. We used to think that this kind of plasticity was limited quite stringently by a critical period in early life. But we now know that it's possible to observe plasticity in these somatotopic maps even into adulthood. And this was shown rather dramatically by a series of experiments led by Michael Merznick at the University of California in San Francisco in the 1980s and in years since then. And many experiments have led to similar kinds of observations. And here is an experiment that was done on an owl monkey. And the experiment involved the surgical removal of a digit in the contralateral hand, following a mapping of the somatic sensory cortex. And so the question was, how would that somatic sensory cortex change in the months following the surgical amputation of a digit? And so here on the left-hand side of this figure is a map of the hand region in the somatic sensory cortex in area 3B and adjacent area 1. And what we see are the territories that contain neurons that were responsive to stimulation of each of the 5 digits on the monkey's hand. And then following the amputation of digit number 3, what was seen after several months of recovery is that the cortical area that responded to the digit number 3 before the surgery has now come to represent the adjacent digits 2 and 4. So now this part of the brain seems to have been taken over by circuits that were either already there and silent or represented the growth of new synaptic connections into this region of the hand representation. Well, I would invite you to refer to an interesting box in the textbook, if you have a copy of the textbook, that considers the phenomenon of phantom limbs. Phantom sensations generally we now think are generated at the level of the cerebral cortex, and it reflects this kind of plasticity that follows injury to a nerve, whether or not there's actual removal of a body part. Well, this is a subject that I don't mind telling you that I've come to know quite a bit about, not of my own choosing, but because of a condition that I developed some years ago that required the surgical removal of a portion of my right jaw. I'm not sure if you've noticed that I tend to be a little bit weak here on the right side of my face, maybe just slightly asymmetrical. It's because the right side of my jaw is now comprised of titanium and hip bone. And in the process of undergoing surgical interventions for a condition that led me to this state, I've had severe damage to the mandibular division of the trigeminal nerve. So this has led to the experience of phantom sensations around the region of the right side of my face and into the oral structures. And over the years, they have changed somewhat, and I suspect they reflect something like this process that's being outlined here, where there is now invasion into a region that has been deafferented by this surgery. And in my case, it reflects the cutaneous surfaces as well as the oral surfaces on the right inferior portion of my face. Well, sometimes these phantoms are a bit distressful, but oftentimes they are somewhat entertaining, I suppose. Like the man who knew too much in the Hitchcock film, having some information in mind can both be troubling and also at some times somewhat entertaining. Well, enough about me. Let's get back to the subject of cortical plasticity in life following some kind of modulation of body structure or function. Well, one does not have to have amputation or nerve section in order to induce plasticity in the regions of our sensory and motor maps. We know this from experimental studies in animals and now studies in human subjects, where it's possible to relate the relevant map to some particular experience of the body. And in this case, monkeys were subject to cortical mapping in order to represent the distribution of receptive fields in the somatic sensory cortex as they relate to each of the 5 digits on the contralateral hand. And in this case, monkeys were trained in a task that especially activated digits 2 and 3, and to a lesser extent digit 4. And after some period of differential stimulation of these digits, what we see is a fairly significant expansion in the amount of the somatic sensory cortex that's representing these 3 stimulated digits, perhaps at the expense of the adjacent representations of digits 1 and 5. So this is generally the case for all kinds of different sorts of manipulations. For example, it's been studied in the somatic sensory and motor maps of violin players that typically the right central region of the central sulcus cortex is enlarged structurally and functionally compared to the corresponding region in the left hemisphere. And if one considers the act of practicing the violin, I think we gain some insight as to why that might be the case. Consider the fingerings of the left hand along the neck of the violin. It requires incredible skill to fractionate the movements of the digits of the left hand. Perhaps a qualitatively and quantitatively greater amount of skill and experience than does it take to bow with the right hand if one is playing the instrument in the conventional way. Well, when the somatic sensory and motor maps of these violin players have been studied, again, one sees evidence for an exuberant growth of the corresponding region in the right central sulcus compared to the left, perhaps reflecting the kind of neurobiological change that we see illustrated in this experimental study using this non-human primate model. Well, what kind of mechanisms then might be responsible for these kinds of changes in the somatic sensory and motor maps? Well, hopefully you will recall the mechanisms of synaptic plasticity that we discussed in Unit 2. That would include long-term potentiation, long-term depression, the consequences of these physiological changes for the growth or the pruning of synaptic connections. And one might imagine that in this particular case, where we see an overall expansion in the dimensions of the cortical areas that are representing the experience functions, we imagine that perhaps long-term potentiation has been especially prominent, leading to the growth of new synaptic connections along dendrites. So if indeed that has been the case, this would occupy more volume and potentially account for these larger representations. Well, what kind of circuits in the cortex might actually be responsible for these kinds of synaptic changes that might lead to an expansion in cortical circuitry volume? Well, I would suggest that one interesting circuit arises from these pyramidal cells that we find in the upper layers of the cortex. Now, I will remind you that the thalamic input comes into the middle layers of the cortex, and then from there these stellate cells and small pyramidal cells relay information up to cortical layers 2 and 3. And in cortical layers 2 and 3, we see the computation of new functional properties, but we also see the extension of long-ranging connections that allow for one cortical column to communicate with nearby columns within the cortical area, and even to communicate with distant columns in adjacent cortical areas. So this computational function of the cortical microcircuit, together with the dissemination or the communication of these signals to other cortical columns, is an important function of these pyramidal cells that we find in the upper layers of the cortex. Well, in studies that my colleagues and I have done, looking at the organization of the cerebral cortex, we've had occasion to study these long-ranging connections that are grown out by these cortical pyramidal cells in the horizontal dimension. And the way we've typically studied these connections is by making an injection of a tracer substance into a local volume of the cerebral cortex above cortical layer 4. That substance then is taken up by the neurons that are present at the site of the injection, and the tracer substance is transported along the axons that grow out from these cells. And what I'd like to show you is some data from my own lab that I generated with the help of my colleagues that illustrate this cortical network. So we're looking at a tracing of axons following one such experiment where the injection of the tracer was made right here in the middle. It's as if we're now looking down upon the cortex from above. And so this is where we would find the cell bodies that have taken up the tracer. And all of these short line segments that you see represent the axons that have grown out away from these cells. There's a bit of a halo here in the middle where I did not trace the axons because they are simply so dense close to the cell bodies. It's difficult to differentiate the labeled axons from the dendrites at that point, so I simply omitted them from this tracing. And what we see then is that these axons extend for several millimeters across the cerebral cortex. And I'll just remind you that each hypercolumn within the cortical circuit is about a millimeter or so in diameter. That's probably actually an upper boundary for the size of a human hypercolumn. We don't quite know precisely what it is, but it's somewhere a millimeter or less. So what we see is evidence of connections that are extending well beyond the typical hypercolumn. And the implication is that these are connections that are connecting multiple hypercolumns together. And so these connections provide then for a substratum that potentially might mediate plasticity in the brain. Well, let's take a closer look at these data. I'd like to actually show you what these cells look like. So if we were to focus in now on a region of the injection site right in the middle, here's what we would see. This is a dark field photomicrograph showing you a cluster of about a dozen or so neurons. And what we're looking at is mainly the cell bodies, these small little brown spots, and their dendrites, these thick tufts that are proceeding out from these cells, and then these finer thread-like structures. Here's one right there. There's another one over here. These are these axons that are extending over some distance across the cortical mantle. And if we were to look out in a more remote site, for example, out here, what we would see is a terminal cluster. So all of these bright little line segments here, these are all portions of axons where we have terminals of the axons that arose from cells that were several hypercolumns removed from the location where we find this terminal cluster. Now, the anatomy is interesting in and of itself, but what makes this really fascinating is to relate the anatomy to the physiology, and we can do this in this particular study because we made injections into the visual cortex after we mapped the distribution of columns that are representing different orientations of visual stimuli. And what we and actually several other groups ahead of us have done in other model systems is demonstrated that there is a principle of like connecting to like when it comes to these long-ranging horizontal connections in the cerebral cortex. And what I mean by that is that if one were to inject, let's say, a region of the visual cortical map that has a preference for near vertical orientations, what we find is a clustering of connections that has a preference for overlying other columns that likewise represent near vertical orientations. And so this now suggests that what we're seeing here is a capacity within this cortical network to represent different regions of visual space that are responding preferentially to the same stimulus. Well, why am I telling you this about these horizontal connections in the cerebral cortex? It's because this horizontal network very likely mediates the plastic reorganization of functional maps in sensory and motor regions of the cerebral cortex. We know this from a variety of studies that had looked at how does this horizontal network in the somatic sensory cortex respond to nerve injury or to immobilization of a joint that alters the way that that musculoskeletal unit can be used by the animal. And in such instances, we see really a tremendous amount of exuberant growth within the cerebral cortex involving these kinds of horizontal connections. And it suggests that there is a dynamical sequence of events that must take place following the perturbation that leads to the growth of these connections. It presumably involves perhaps the awakening of silent synapses that are already present in this network, and that might then be followed by the growth of new axonal collaterals that extend the range and the density of synaptic connections. Now, we'll say more about the growth of axons in the brain, or lack thereof, following injury to the brain itself. But at least with injury to peripheral structures or to the structured use of the peripheral structures, we think that this horizontal network is an important mediator of the adaptive plasticity that can follow in the consequence of that kind of disturbance. Before we talk about the response of the brain to direct injury of itself, let's just touch briefly on the mechanisms by which peripheral nerves respond to injury. Well, this is the good news part of the story of regeneration of nervous tissue. With damage to a peripheral nerve, the axon that has been injured has significant capacity to regenerate and to reestablish a functional connection with the target of that axon. So typically this is a process that unfolds in a sequence. Following an injury to a nerve, so for example, we imagined there may be some kind of surgical or traumatic injury to a peripheral nerve, be it a spinal nerve or a cranial nerve, and what we see is a degeneration of the distal portion of the axon. However, the entire nerve typically is not lost. Rather, the macrophages invade the region and consume the debris in the wake of the injury, but the Schwann cells then will proliferate. And the proliferation of these Schwann cells basically establishes a channel through which a regenerating axon might locate its target and reestablish a functional connection. But along the way, the Schwann cells really are the key mediators of regeneration and regrowth of this injured axon, because the Schwann cells begin to produce growth-promoting substances. These growth-promoting substances can influence the expression of growth-related genes in the cell body of these neurons. And that can then induce the formation of a growth cone, which can then respond to local signals that are produced by these Schwann cells and laid into the extracellular matrix. And thereby, a growth cone can be reestablished, it can become motile, and it can recognize adhesion molecules, and it can respond to other kinds of inductive signals that are produced by these Schwann cells that are now proliferating and reestablishing this channel through which this growth cone can eventually find its peripheral target. Now, once the growth cone does make it to the target, be it a muscle fiber or a ganglionic neuron, in the case of a preganglionic visceral motor neuron, for example, we have the capacity to reestablish a synaptic connection. Now, this typically is not done with perfect fidelity, so that the connections that are reestablished are never 100% of what was there previously, and often the rules that shape the connections in the developing embryo are recapitulated, but perhaps not with the exact degree of precision that was once possible. So as a consequence, one typically doesn't see full functional recovery with peripheral nerve injury. I know in my case, I have had some return of sensation in the cutaneous surfaces, but I remain somewhat impaired in certain motor functions involving the lower right regions of my face. So at least following my injury to the mandibular division of trigeminal and some branches of the facial motor nerve, I'm still awaiting the restoration of full function, and of course doing everything I can to rehabilitate this process. But nevertheless, I think the realistic expectation is that the process of reinnervation at best will be incomplete. Now we are talking about the changing brain across the lifespan, and unfortunately as we age, the likelihood that we will encounter some kind of an injury that changes the structure or the integrity and the function of the nervous system is increasing. So we're going to turn our attention now to the way that the central nervous system responds to injury. And as we'll see, despite its best attempts to do so, there are active mechanisms that limit the regenerative capacity of the brain and the spinal cord following injury or disease. Well, there are essentially three ways that the central nervous system can be injured. It can be injured through trauma, it can be injured through hypoxia or ischemia that denies the brain of the oxygen or the blood flow that's necessary for full neural functioning, and the brain can degenerate under the conditions that are consistent with a neurodegenerative disease. In each of these three categories of central injury, there is often the acute risk of the loss of neurons right at the time of injury. And this happens basically because of two considerations. The first is excitotoxicity. And as you may remember from Unit 2 in the course, excitotoxicity refers to the destruction of neurons due to the release of glutamate and the interaction of glutamate with its receptors that set off a sequence of events that leads to the lysis of a cell. So neurons can be excited to death by an elevated concentration of glutamate in the extracellular fluids. Well, this can happen during acute injury to the nervous system because as brain tissue is damaged, the glutamate that's found within cells is now released to the extracellular fluids, but also there is typically an increase in electrical activity, which leads to an increase in the synaptic release of glutamate. So the direct release of glutamate from damaged cells together with the synaptic release of glutamate elevates this neurotransmitter to levels that interact with the receptors on surviving neurons to the point where dangerously high levels of intracellular calcium can become established in the postsynaptic neurons. And with dramatically high levels of intracellular calcium, well beyond what is physiological for inducing long-term potentiation, for example, these pathologically high levels of calcium can trigger a sequence of events that leads to the programmed cell death of neurons. This is a phenomenon that we call apoptosis. And we know more now about what these pathways are. So as calcium enters through typically NMDA receptors for glutamate, as well as other signaling molecules related to damage to cells, to hypoxia, to stress, even to the withdrawal of growth factors following injury, these factors can interrupt the normal function of this signal called BCL2. So when BCL2 is blocked, this allows a sequence of events to unfold that leads to the activation of this important enzyme, caspase-3. And caspase-3 is what can trigger this downstream set of mechanisms that leads to programmed cell death. This involves the condensation of chromatin, the fragmentation of DNA, and the disruption of nuclear membranes. Now, let's consider what happens to these neurons that are just on the margins of the injury. So we can consider this to be like the shadowy margins of the infarcted brain tissue following, let's say, a stroke or a traumatic brain injury. In fact, we sometimes call this region of tissue the penumbra. So the penumbra means shadow. The penumbra is a region of brain that has been stressed by the injury, but these neurons have not necessarily been pushed into excitotoxicity or apoptosis, programmed cell death. So these neurons will survive, but perhaps many of their axons have been severed. And the question is, what will happen to these neurons? Do they have capacity to regenerate new connections and restore some functional capacity? Well, there is a mixed answer to this question that we're beginning to understand. And part of the reason why the answer to the question is mixed is that it may depend upon where the axons are growing. The axons that are trying to grow back through white matter have a much more difficult time with regrowth and regeneration than do the axons that are spreading out in the horizontal dimension within gray matter itself, such as the cerebral cortex. So let's first consider what's going on within the white matter, where the axons that might have supplied a long pathway, such as the corpus callosum or the cortical spinal tract, are going to have a very limited capacity to regenerate and regrow a normal axonal connection. So let's think about why that might be the case. So imagine that we have some neurons in the cerebral cortex that are extending axons into some white matter structure. And if there's some region of injury now that affects these cells, so there's some injury to neurons within this region, one of the most dramatic reactions that will happen around the margins of this infarcted tissue is proliferation of glial cells. In fact, the proliferation is so intense that this local volume of brain becomes congested by these glial cells and their processes. And it's not just a physical congestion, but it's also an exuberant production of chemical signals that have a negative effect on the growth potential of the surviving neurons. Indeed, if we look at what happens following an injury within a white matter structure of the brain or the spinal cord, what we see is a rather dramatic reaction, first involving the infiltration of microglia that will phagocytize the cellular debris. Astrocytes become activated and might likewise migrate into the area, and then the oligodendrocytes themselves will proliferate. And together these glial cells will form a scar of tissue that continues to produce substances that have a suppressive effect on the development of a modal growth cone and the activation of growth-promoting programs by this injured neuron. So there are a variety of molecules that are expressed by these glia, some of which are especially important and are expressed by the oligodendrocytes themselves. And among these molecules are integral membrane proteins, such as this molecule here, this OMGP, which stands for oligodendrocyte myelin glycoprotein. And this integral membrane protein, MAG, myelin-associated glycoprotein. And then this intriguing protein that you can read about in the textbook, if you like, called NOGO-A. And what these integral membrane proteins seem to be doing is inhibiting the expression of growth-promoting programs by injured neurons. Now, in addition to these integral membrane proteins, there are secreted ligands that will interact with a variety of receptors that are expressed on the membranes of the axon and a growth cone that may be attempting to regenerate. And these ligands interacting with their receptors seem to promote the collapse of that growth cone and an active inhibition of growth. So one of the problems for the central regeneration of axons within compact white matter within the brain and spinal cord is this active suppression of growth that's mediated via these signals that are expressed on the surface of glial cells. So one might imagine a variety of strategies to try to overcome this, either by suppressing these signals through immunotherapy or other kinds of pharmacological therapy. One might imagine providing a permissive environment that would provide a route for the regenerating axon to avoid this plethora of signals that will actively suppress the growth and elongation of a regenerating axon. Well, there are a variety of strategies that have showed some limited success in animal models. Unfortunately, we've yet to see dramatic success in any human studies, but with ongoing study and ongoing discovery, I think there is potential to develop strategic interventions that will overcome these intrinsic limits on the capacity of central axons to regenerate. Now let's just back up a moment, and I want to consider the neurons that are primarily living now in gray matter and sending out axons in the horizontal direction within the cerebral cortex. Now for these neurons, the story seems to be a bit different. And perhaps it's because in the gray matter of the cerebral cortex, we don't have nearly so much myelin that might be expressing all of these inhibitory signals. What animal model study suggests are the exuberant growth of horizontal networks within the cerebral cortex following local injury. And so this provides a substratum for plasticity and adaptation following an injury to a region of the central nervous system. And I think from a standpoint of rehabilitation, the outgrowth of this horizontal network following injury provides opportunity to shape that plasticity and to provide the kind of structured patterns of pre- and postsynaptic activity that would align itself with Hebb's postulate and our principles of spike timing dependent plasticity that might produce adaptive change, adaptive plasticity that could lead to the restoration of function. So as we're just now beginning to understand these rules of plasticity and the molecular signaling that can promote growth or inhibit growth, I think we have tremendous potential now to develop strategic interventions that combine physical therapy with pharmacological intervention, perhaps even genetic medicine, as a means by which we can optimize the plastic potential of circuitry within the cerebral cortex to overcome the impact of disorder and disease that may be associated with focal injury. Well, lastly, I'd like to talk about the limited potential of the brain to generate new neurons throughout life. Now, I emphasize limited because there was a time, maybe about a decade ago, where there were numerous studies suggesting that the creation of new neurons from stem cell populations might be widespread throughout the cerebral cortex. Well, through a set of rather compelling studies done in the last 5 or 6 years or so, I think we've settled this question, and we now agree that the production of new neurons in the human brain is really limited to a special set of stem cells that seem to sit in the basal region of the dentate gyrus of the hippocampus. So in this subgranular zone, we have a population of neural stem cells that can differentiate into interneurons that are found in the granule cell layer of the hippocampus. So it's very clear that neurogenesis in the adult human brain does not occur over the entire cerebral mantle, but rather is restricted to this small region within the medial temporal lobe. Now, that's not to say that this is functionally unimportant or clinically insignificant. Actually, quite to the contrary. We now know that this genesis of new neurons in the hippocampus can be modulated by all kinds of different interventions, including pharmacological agents such as antidepressant medications, as well as physical interventions such as exercise. And generally speaking, this is a bit premature to say this, but I'll do it anyway. Generally speaking, as measures of health and wellness increase, so we see an increase in the rate of neurogenesis in the hippocampus, at least as predicted by animal model studies. Conversely, those indicators of a reduction in quality of life, be they disease or be they mental illness such as depression or other modifying variables such as inactivity or sedentary lifestyle, these kinds of factors tend to reduce the rate of neurogenesis in the hippocampus. Now, I don't think anyone really understands why generating new neurons in the dentate gyrus of the hippocampus ought to be associated with health and wellness. But if indeed it is the case, then I think this is actually good news, because it suggests that there may be multiple mechanisms that are available to most of us by which we can optimize neurogenesis in the hippocampus and thereby promote healthy brain function and health and wellness more generally. So, with that said, I look forward to an opportunity to exercise later today or first thing tomorrow morning and maybe inspire a modest increase in my rate of hippocampal neurogenesis. Okay, well, this concludes Unit 5 of the course. So we've now explored mechanisms of neural signaling, we've surveyed our sensory motor systems, and we've had a chance to discuss brain development in early life and how the brain changes across the lifespan. What remains in our course is an exploration of the more complex brain functions that will give us an opportunity to really synthesize and integrate everything we've been learning in medical neuroscience, including the anatomy of the human brain. So I really look forward to getting into the final unit of the course and having these discussions that I really hope will put a bit of a capstone on your experience with me in your studies of medical neuroscience. So I'll see you on the other side of the Unit 5 quiz and look forward to speaking with you in Unit 6 on complex brain functions.